Good morning, everyone, and welcome to this session that we are starting now. Just a few words to introduce myself. My name is Giulia Zori, and I work as a researcher at the National Research Council of Italy. And I'm very happy to be here, and thank you for the organizing committee for asking me to chair this session. Just a few words, but I don't want to take any more time because we are already a bit late. So I give the floor immediately to Professor Rick Jelen of Brigham Young University in Provo, USA, who will talk us about the exotic halophyte resources for improving quinoa. And I'm very interested about what he's going to show after the panelist discussion that started yesterday. Thank you very much, Professor. The floor is yours.
Thank you very much, Julia. It's a pleasure for me to be here again, and especially to share work that I've been doing for the last 25 or so years on quinoa. My close collaborators at Brigham Young University, or BYU, are David Jarvis and Professor David Jarvis and Professor Jeff Maughan. So I just wanted to make sure I acknowledge them, as they've been heavily involved, particularly in the genomic part of the work that I'm going to show you. We have developed over the years a number of close collaborations, including Mark Tester here, and we tremendously appreciate the contributions that they have made and definitely want to recognize their role in this work. I should also note that when I started working on quinoa in the year 2000, my interest in quinoa was really triggered through a number of different things that happened at the same time, one of which was a trip here to Morocco. It was the first trip I made to Morocco, and I had been connected with Professor Wafa Ben Habib of the IAV. And Wafa and I quickly became really, really good friends. We came to look at wild oat, avena, genetic resources. That's my other life that I have, is working on avena genetics and genomics. But Wafa and I were talking about, as we went around, particularly northern Morocco, and looked at the drought situation. Farmers were turning away from leguminous crops, sources of protein in their diet, and we thought of quinoa as a potential substitute with its climate resilience. And so from the next year on, we began working to introduce germplasm and work with farmers, particularly in the Hanifra region, to identify whether or not quinoa would be adaptable within the, at least in the northern Moroccan, central Moroccan environment. And we're very pleased with the way those results worked out. I just shown you some of my other collaborators. Most of them are in universities in Peru, Bolivia, Argentina, in South America, and then in sub-Saharan Africa. Places like Malawi, Professor Moses Maliro here. And then my basic science colleagues, I'll be showing you some results of some basic science work that we've been doing with Professor Tester and with a group of very strong botanists, particularly in Eurasian Chenopodium at the Czech University of Life Sciences in Prague. So our goal when we started, we were working with almost a neo-domestication situation. We had a crop that had been domesticated 3,800 meters above sea level in the Andes of Bolivia and Peru. And we were taking this crop, and there were strains of quinoa that were adapted already to a Mediterranean environment in the central Chilean coast. And we were working primarily with local knowledge, okay? And local knowledge is great, but local knowledge we have to at some point tie that local knowledge in with hard scientific data. And one of the things that we did not have a clue about was what are the genetic resources for breeding quinoa. We knew some of the primary genetic resources, the vast majority of which we as citizens of the United States of America, as non-signees to the international treaties of the 1990s, were not accessible to us. And so we had to work with potentially a relatively small pool of germplasm that was available through, that had been exported from the Andean region prior to the year 1993. And so this raised a lot of different questions. We got into the taxonomy of Chenopodium and found that it was a complete and utter mess. This is one of the most difficult genera taxonomically. And so we initiated work to try and understand first the molecular structure at the chromosomal and DNA level of quinoa, and then started expanding those studies to wild relatives to try and figure out what the secondary and eventually the tertiary gene pools are for improving this crop. And for adapting this crop, which we saw as potentially, as I mentioned yesterday, potentially as a staple crop that could feed much of the world with a highly nutritious staple, better than rice, better than maize. So we proceeded that way. This is kind of where we are in terms of the life history of quinoa. I think there's a pointer on here. Yeah. So now we know quinoa is an allotetraploid with 36 chromosomes. We know its genome composition. It contains the A and the B subgenomes. The A subgenome, the closest species to the original donor that we have, is a species called smooth goosefoot, is the common name, or Chenopodium subglabrum. We know that Chenopodium subglabrum, from sequencing the mitochondrial and chloroplast genome, was the maternal ancestor. It was the pollen recipient. The pollen donor was a B genome diploid. What's interesting about the B genome is the B genome is, the botanists tell us, is not native to the Western Hemisphere. Although now we think we have botanical specimens that our Czech colleagues have identified from Alaska that are Chenopodium suecicum. Interestingly, suecicum, or Swedish goosefoot, is the species that is most closely related when we compare whole genome sequences from the B subgenome of quinoa to the existing B genome diploids. Kind of interesting, there are only 3 B genome diploids that we know of. Swedish goosefoot, Chenopodium suecicum, fig leaf goosefoot, Chenopodium fiscifolium, and Ukrainian goosefoot, Chenopodium ukrainicum. And but the B genome is found in a number of different Eurasian and New World, one New World polyploid, you see it here, quinoa, and a number of different Eurasian polyploids, including lamb's quarters Chenopodium album, which is our terrible weed. These two crossed a number of years ago, somewhere in the 1 to 5 million year range, roughly. And the product of that hybridization in the wild was, after chromosome doubling, was a stable allotetraploid we know now was Chenopodium berlandieri, or pit-seed goosefoot, that Mark talked about yesterday. This species, I'll show you something about its ecological distribution. We are particularly interested in pit-seed goosefoot because the United States of America is the center of origin and therefore the center of diversity of pit-seed goosefoot. It's ironic quinoa was domesticated thousands of kilometers south in the Andes Mountains, and yet the center of origin of the polyploid is in North America. This, we believe, was transported probably epizoochorically in birds to South America, where it became established as classified as a separate species, but biologically it's the same thing. Chenopodium hircinum, or avian goosefoot. And avian goosefoot was domesticated in the Andes to give us quinoa. What's also interesting about Pittsy goosefoot is Pittsy goosefoot was domesticated at least twice in antiquity. It was domesticated anciently in Mesoamerica, primarily as a vegetable. The panicle is cooked. The flowers are stripped off after boiling or steaming. They're made into patties, coated with a coating, and then fried, and then wrapped in a tortilla with cheese and hot sauce. And it's a common street food in Mexico City and cities around that part of southern Mexico. The other domestication event of Pittsy goosefoot happened in eastern North America around 6,000 years ago, according to the archaeologists. And that plant, that crop, as a seed crop, was eventually displaced by maize, beans, and squash, okay? By the cucurbits and by bean, common bean, Phaseolus, in eastern North America, sometime around or slightly before the arrival of the Europeans. So here is our latest attempt at deriving the family tree, a picture of the primary and secondary genetic resource pools for quinoa. You can see down here, this cluster represents cultivated quinoa. This is looking at approximately 13,000 SNPs that have been screened and corrected for ascertainment bias against a quinoa and a Mexican quinoa reference genome. From this phylogenetic tree, a maximum likelihood tree, you can see clearly the two domestication bottlenecks. One in Mexico, this is nut, stands for Chenopodium berlandieri, subspecies nataliae, and at the base of this cluster, we have 3 accessions of Chenopodium hircinum from South America. Okay? So what I want to impress upon your minds is the tremendous range of genetic variation that is present in the 1, 2, 3, 4, 5, 6, 7, probably 8, 9, 10, 11 different ecotypes of Pittsy goosefoot in North America, with the additional ecotype pending further investigation of the South American material, which is ongoing with Mark Tester's lab, trying to define the South American material. But there's all of this genetic variation in North America, including this. And this is a conference on Biosaline Agriculture. And I just want to show you, this is the Atlantic Ocean. This is the high tide line, and this is a plant of this ecotype, the Macrocalicium ecotype, growing along the high tide line, being fertilized by decaying seaweed, decaying algae along that high tide line on the coast. A little bit more of that. So here is a picture of the ecotype structure of wild quinoa in North America. And again, we have one ecotype that we've identified in South America that includes both wild and cultivated types. Notice there are 2 ecotypes that are deeply associated, closely associated with proximity to the ocean. There is one ecotype that is found in the hot deserts of the southwestern United States, many of which are composed of saline or sodic soils. And then there is at least one sub-ecotype of this Zishakii, we call it ecotype, that is found in the endorheic basin called the Great Basin of the western United States. There's Utah, where my university is right here on the eastern edge of this large area known as the Great Basin. It's a divergent rift zone, and I'll show you some interesting genetic resources from this area. So we've made an extensive collection at BYU, not only of tetraploids, AABB tetraploids here, but also of diploid genetic resources of the tertiary genetic gene pool. And just a picture of, these are not actual readings of soil salinity, but each one of these bolded 14,111, 1465, 937, 1821, each one of these represents a different accession of the Bassianum ecotype that is closely associated with either saline bayous or beach sands growing right along the ocean. And you can see the salinity measurements. These are from National Oceanic and Atmospheric Administration, NOAA data, okay, of oceanic salinity. This is the Gulf of whatever here, okay? And here's a photograph, a highly diverse population of Chenopodium berlandieri variety Bassianum. This is the Gulf of Mexico. Okay, this is the open Gulf of Mexico. Here is another population that's growing. This is a saline bayou along the delta of the Mississippi River in the state of Louisiana. Here is a population of plants from a cross that we've made between a lowland Chilean background quinoa with one of these Bassianums. We've used these Bassianums a lot as crossing partners. This is why, because these are data from plotting linkage distance on the y-axis against the linear sequence of each of the 18 chromosomes of quinoa. And what we see are recombination maps that align perfectly with what we would expect, almost perfectly, if we have two different strains of the same biological species. They're able to cross, they're able to recombine. We have a good distribution of recombination. As expected, we have flat zones where there's very little recombination around the centromere of the chromosome. This is typical. Okay. We do see one small rearrangement in this cross. We think this might be a real rearrangement, a terminal inversion. And I'm not going to talk about the significance of that, but there could be some significance. That's on chromosome 4A. There's another inversion here, a large inversion on chromosome 3B, which together with Mark Tester's lab, we discovered is found primarily in lowland Chilean quinoa germplasm. Now this was a cross with a highland Bolivian quinoa, but we plotted it against the sequence of our reference genome, which is a lowland Chilean reference genome, which carries this inversion. So this inversion is not affecting this cross, okay? It's only affecting a cross if we were to use lowland Chilean parent that carries that inversion. It's about 20% of lowland Chilean quinoas. Here is just a close-up of a couple of these restriction maps, the one for chromosome 3B showing the inversion. Here's chromosome 2A. If we compare the linkage map of North American by Bolivian, wild North American by cultivated Bolivian quinoa, if we compare that linkage map with the South American wild, 1101 from Argentina, to cross to the same quinoa parent, we see comparable linkage maps. Okay? The only thing, the linkage maps for the North American by quinoa cross are compressed by about 10 to 20%. There's about 10 to 20% less crossing over. But the distribution of crossing over is the same. What this means is that breeders can use this material right now to intergrass or to try and introduce wide swaths of the genome into quinoa, and breeders can expect good recombination. Okay? So what we're saying here is that geographic proximity is not an indicator in quinoa of how well that strain is going to cross with a South American quinoa. We can use materials from North America that may be even closer or just as close genetically. Here's the difference geographically between 1101 and Real. That was the bottom linkage map that I showed you. And then 937 by Real was the top linkage map that I showed you. We think this may be a reflection of long-range dispersal by birds along the Central Flyway to South America. We also have a reference genome that we published last year, again with Mark Tester's group. We have a reference genome for Mexican domesticated quinoa. It turns out that Mexican domesticated quinoa is more different from 937 than 937 is to either QQ74 or Real 1. There's an additional inversion in the Mexican material on chromosome 4b, if I remember correctly. So here is another ecotype. This is the ecotype that grows along the high tide line. You can see again what the oceanic salinity is like from NOAA data. This is what the habitat of this species looks like. This is growing at the high tide line along Periwinkle Cove, coast of New Hampshire. This ecotype has very large seeds. The seeds are roughly the same size as cultivated domesticated quinoa. And when we make the hybrids, we can see heterosis, a little bit of heterosis or hopefully complementary gene action, which there may be some room there to select for larger seed size, as well as possibly tolerance to salinity. And so here's the southwestern ecotype and different soils that this grows on. Segregating population with the southwestern desert ecotype. Here's the Great Basin ecotype. And this is a picture of the habitat. Here's one plant growing, a lone plant here in the middle of the Nevada desert. This is a very sodic, somewhat saline, but very sodic environment. Here is a population of a different ecotype in South Texas, growing on a vertisol, shrinking, swelling clay soil that is highly affected by salinity. And just another population here. Here is a high plains ecotype growing on a saline sodic soil, 8 to 16 millimoles per centimeter salinity with a sodium absorption ratio of around 30. So just to show the relative fertility of the F1 hybrids, there is variable fertility depending upon which parental combination we make. And I just wanted to show you, these are advanced lines that we've derived selected from crosses in these wide cross combinations. You can see the seeds are large, that's 2 millimeters in diameter. We can select out material that is just like quinoa from these crosses. It takes a lot of work because the domestication character in many of these crosses is segregating 1 to 15. So we can identify material that is higher yielding. In Peru, this is a Peruvian desert environment, saline soils. This is the control check, Blanca de Juli, and advanced lines that exceed Blanca de Juli for seed size as well as for yield. And I wanted to just show you my last slide. The tertiary gene pool is even more interesting. This is a black alkali soil in western Nevada, and this is Chenopodium nevadense, an A genome diploid. We've made several collections of this species, and this is a species that apparently not only tolerates alkalinity, but also potentially flooding within the root zone, which quinoa geneticists and breeders well know is it's not something that is a typical characteristic for quinoa. So thank you for your attention, and it's been fun.
Thank you very much. Really interesting presentation. I would give the floor to the next presenter and leave some minutes in the end for questions, if you don't mind. So now I would like to call Kaola Lariki. Please. Thank you very much.
Hello, my name is Khabla Al-Harishi. I'm a PhD student, and I'm doing my research at the University Mohammed V in collaboration with the International Center of Agricultural Research in dry areas. I'm pleased to be here with you and share my research on stem resistance in elite spring breed wheat genotypes. And the presentation will include some preliminary results focused on adult plant growth stage. This is to support the development of resistant varieties. So breed wheat is the most consumed cereal. It's provide up to 70% of cereal intake, and in urban areas, and up to 66% in rural areas. It contributes also to 60% of daily intake and up to 40 of protein intake. But the production of wheat in Morocco is under serious threat. Actually, in 2024, there was a drop of 40.6% compared to the year of 2023. And this is mainly due to abiotic stresses like heat and drought, and biotic stresses like disease and pests. So, among the wheat diseases that are really severe in Morocco, we found yellow rust, septoria leaf blotch, stem rust, and leaf rust. And for their development, it's essential to have the environmental conditions and to have also the pathogen. And for the pathogen to occur, it's really important to have some environmental conditions and also to have a susceptible variety. So the diagram presents the three factors that are essential for the disease to occur. I will focus on the wheat rust. They are called the cereal killers. Among them, we found yellow rust caused by the agent Puccinia striformis forma specialis tritici, leaf rust caused by Puccinia triticina, and stem rust that is caused by the pathogen Puccinia graminis tritici. And each one of these pathogens requires a special temperature and especially the condensation of the moisture on leaf surface. Actually, the global annual of yield losses caused by rust are up to 15 million tons. Stem rust alone can cause 6.2 million tons. In Morocco, yellow rust is the common disease and can cause up to 50% yield losses. But in the recent years, stem rust is re-emerging and causing a serious threat. So it is important to know the life cycle of the Puccinia graminis. It is an obligate biotroph. It means like it's essential to survive for it, a plant tissue, and it's a heterogeneous species. It requires two hosts, a primary host which is the wheat. It undergo its asexual cycle. And an alternate host, which is barberry, which undergo its sexual cycle. Actually, in Morocco, barberry is really rare. So what we see usually, it's like a clonal population of sexual recombination. So it is important to know the life cycle. To disrupt the disease resistance and to understand this resistance. So over 60% of over 60 stem rust resistant genes were identified, but most of them, they have lost their effectiveness, and this is due to the virulence of some races. In Morocco, several stem rust races have been identified, including TK-TTF, which caused the epidemics in Ethiopia in 2013 and 2014, and it was confirmed in 2019. And also we found TTRTF along with TK-TTF and TK-TTF that reflect the emergence of stem rust in the Mediterranean region. These stem rust races pose really a significant threat, and they are causing like real damage in wheat production. So for the stem rust resistance, there are two types of resistance. The old stage resistance, that are the seedling stage resistance, and the adult plant resistance. Actually, the seedling stage resistance, it's really it's force, but it's life shorted. But for the adult plant resistance, it's like more durable and have broad resistance. So we don't have to rely on the seedling stage resistance, but we have to combine the seedling and adult resistance to have like more durable and resistant varieties. So in Morocco, according to a study that have been done last year, we found that there is some effective genes. There is still some effective genes in Morocco, like SR2, and SR24. SR2 it's durable and have adult plant resistant, and some ineffective genes like SR5. So the aim of this study is to assess the genetic diversity and the population structure in elite spring breed with genotypes, to evaluate the phenotypic variation, also to identify significant association genomic regions with GWAS, and finally to predict genomic breeding values using some models. So to enhance this resistance, first we have been working on 384 elite genotypes. We have been doing the evaluation in 2 environments in 2024 and 1 environment in 2025. And this environment is CDLLTZ, which is a hotspot for disease. After this, we have received the genotyping data using the 25K weight array. And we combined the severity data with host reaction to calculate the coefficient of infection. And finally, we have used, to analyze the data, we have been using some models. So in this slide, we will see, you are seeing how we have been scoring in the field. So we have a scale from 0, that is immune, to susceptible, and it's this scale. It's it's especially about the size of the Uridina spores, and also for the necrosis and chlorosis. We have also the severity type. And to calculate the coefficient of infection, we have combined the reaction type with the severity type, and we have got from 0, the response from 0 to 1. 0 is resistant, and 1 is fully susceptible. We have been using 2 approaches, GWAS, which is nothing than a correlation between the phenotypic and genotypic data to identify the QTLs and the gene of resistance. And we have been using the genomic prediction using the GBLAP model run in BGLR package. We have been using the phenotypic data and the genotypic data. And for the results, as you see, we have been some genotypes have been reacting as a low to moderate low to moderate coefficient of infection. However, few of them have highly susceptible reaction. And as you are seeing, this it is skewed to the resistance, and we may have, this is an indication that we may have some adult plant stage resistant genes. And also we have been done the genetic structure population, and the structure shows that there is 3 genetic subgroups. And also the Evano method confirms K3 as optimal. And the structure is used as a model to correct false stratification and reduce false positives. Here are the SNPs that are highly significant. This is the results of GWAS. As you see, these red peaks are the highly significant markers. As you see, we have more significant marker on chromosome 3b, and 5 significant SNPs located on the chromosome 3b are between 765 and 770 megabase, and they are likely tagging the same QTL region. So it may be a strong candidate locus for rust resistance. And we were able to identify another marker that have 24 megabase on 3B also, and it's likely near to SR2, that is a durable resistant genes. And also the SNP located on chromosome 7b at 591 aligns with the location of YR59, for the it is a yellow rust resistant genes. Additional markers on 2D and 4A, 4B, 3D, and 1B may indicate novel QTLs for further investigation. For the genomic prediction, we used the GBLAP model, and this is to know how it's accurate to use only genomic data to predict the stem rust resistance. So we have a an accuracy of the 0.50. That means that 50% of the variation in resistance can be predicted using only data, genotypic data alone. And this is valuable for the breeding programs and can be used for future breeding to accelerate the cycle, and because breeding is time consuming. So to conclude, several QTLs for adult plant stage were identified. These QTLs explain moderate phenotypic variation and may represent both known and novel genes. And the genomic prediction showed moderate accuracy, supporting its use in the breeding program. And after this, we have to validate these QTLs and for their use in marker-assisted selection. And it's essential to have, and it's for further studies, I will be doing more work on seedling resistance and to combine the adult stage resistant with the seedling stage. And in order to pyramid these genes for future breeding programs. Thank you.
Thank you very much also for taking care of the time. I will now introduce Mariam Bibar as the last speaker and postpone the questions after this last speech. Thank you.
Good morning, everyone. So my name is Mariam Gabeber. I am a PhD student here at Asari UM6 Laayoune, and I am very happy to present today our works. So here in Asari, I am working on phenotyping and genotyping of cereals and leguminous. And as a part of our research and work here, I will present our work, which is about the effect of fertilizer amendments and deficit irrigation on physiological and genetic expression of forage sorghum growing under saline conditions. So as we all know that cereal is very important for our life, and they play a very important role as a source of energy for livestock, but the production of cereals faces a lot of problems and challenges like drought and salinity, heat, weed, and other problems. And here in this region, where we are, Laayoune, Sahara region, so the farmer, there is a huge demand of forage crop by the farmer to feed their animals. That's why there is a necessity to introduce some new forage crop and cultivate it to respond to this huge demand of forage crop. So as to respond to this problematic here, we target to work on forage sorghum as a promoting crop in this region here, because first of all, it has a high photosynthesis efficiency and a high sugar content and a high water retention content, and also it can tolerate to a high salinity level. The sorghum grain is a very strategic gluten-free for a food security system, and also there is a lower production of forage sorghum here in Morocco compared to the other world. So to respond to this problematic, we set up our main goal, which is the introduction of high-yielding sorghum tolerant to salinity and drought stress by adopting good agriculture practices. And under this main objective, we set up three sub-objectives. which is the first one is to identify the best fertilizer amendment for high sorghum yield under saline condition. The second sub-objective is to discuss the physiological mechanism of sorghum performance under drought saline condition. The third one is to assess the genetic mechanism behind salt tolerant performance and dry mature. So to achieve these goals, we set up this experiment. So we plant our sorghum in two different fields. The first one in medium salinity and low salinity, to do a comparison between the two sites to see how the sorghum can perform in the two sites under two different levels of salinity. And as data we collected the agronomic yield indicators, and we did also physiological parameters, biochemical parameters, and also we proceeded with the molecular biology analysis. So as a setup of the field, so we start first of all by the preparation of organic and mineral amendments that we used during our experiment. Then after that, we do the installation of irrigation pipe. We did some soil analysis to see the amount of nutrients in the soil. Also, we do the planting, and we did the weed control and the pest controls. So now for the results part, we will start with the agronomic response. So here we have the data of the two platforms, medium and high. And as we can see here, the highest germination and leaf area and also height was recorded under the application of manure and NPK and NPK. For the dry mass, we can see here that all amendments enhance the dry mass in the both sides, medium and low, and also under the two level of irrigation. But we can see here that using the manure and NPK give us more production and more yield production of sorghum in the both in the two sides. For the forage analysis, we can see that all the amendments enhanced fiber, fat, and moisture and ash under the full irrigation in the both sites. And under deficit irrigation, we can see that protein content increased with the application of manure and NPK. For the physiological parameters, here we have the results of RWC, relative water content, electrical leakage, EL, transpiration, and the carbon assimilation. And as we can see here, the NPK reduced the EL under both fall and deficit irrigation. And also we can see that under full and deficit irrigation, manure and NPK show higher RWC. For the transpiration, we can see that manure and NPK and combination of them showing highest value under full irrigation, and the same for carbon assimilation. For the biochemical response, here we have the results of MDA, SOD, and POD and catalase, and we can see that compost show slight high SOD and catalase activity under deficit irrigation, and MNPK show the highest POD under deficit irrigation, indicating a strong stress adaptation. For the molecular biology analysis, so here we have the results of here we can see that we are in the region of Sahara and we are facing the problem of salinity and drought. So that's why we target to work on the genes that are related to the salinity and can help the plant to resist to these problems. So here we choose to work on the SOS. So for the applications for the SOS, we can see that under different treatments, So the gene was upregulated under NPK in medium salinity, and manure and NPK under low salinity in the both system irrigation system. As we say before that, so here we are. We want to respond to the demand of farmers for forage crops. So we are interested also in the biomass. So that's why we target this gene, which is the Rubisco and responsible for the high carbon assimilation. And as we can see here, the overexpression of this gene in medium and low salinity explain the high assimilation rate of carbon dioxide under synthetic and organic amendments, which is NPK and manure. So as a conclusion, we can see that employing the organic amendment, which is manure, and inorganic amendments, which is NPK, or the combination between the two of the two treatments, significantly enhance forage sorghum growth by boosting the defense mechanism of sorghum and make it more resilient in this region. We can see also that the fertilizer amendments have a positive influence on crop production and also the forage nutrition value. We can conclude that the upregulation of Rubisco in the organic and inorganic fertilizer indicates high CO2 assimilation rates. A reduction in its expression of SOS was observed under deficit in the irrigation, indicating that the excessive salinity and water stress impacted the sodium exclusion from the shoot. And thank you.
Thank you very much. We are running a bit late, but we also started 10 minutes late, so I will steal a few more minutes. And first of all, thank you for all the presentations given. I would like to share with you a thought just like this before all the information sediment. So what I do think is that it's really good, as we have seen in the last two presentations, that there are ways to increase the yield of the crops that we are used to know as our staple foods, so both in relation to biotic and abiotic stresses, which is very good, and we shall continue in this direction. But what I also think is that this is not against in adding new staple crops. So if I would be the one to take this decision, I would very much welcome quinoa among our staples, and even many other species. In the last 13 years, I've been working with allophyte, much less known than quinoa, very little known, but that have an interesting potential for a human diet and alimentation. So I will give a talk about this in the afternoon if you're interested to know more. So what I think is that we do rely on good staple crops, but they are really too little compared to what nature has to offer. So I think we should enlarge our horizons in this direction. And we can further discuss this topic at the coffee break. But first, I would like to give the possibility of doing some questions to the speakers. If there is someone who wants to ask something, we can still steal a few more minutes, please. Yes, please.
Okay, can you hear me?
Yes.
So if you allow me, I have 3 questions, one for each speaker.
Of course.
Okay, I will start with Eric. You have showed me this genetic diversity of quinoa, right? And I'm really interested in one major questions about the water consumptions for post-harvest with saponine, and also, defense against pests and interactions with its microbiota. Do you have any clues about that, and you can comment on this variation, please?
Yeah, you can hear me now. Yeah, great question about water, post-harvest water consumption in the processing of quinoa, because quinoa seed has an adhering sticky pericarp that accumulates bitter saponins, those need to be removed. Most of that is commercially removed now through abrasion process, okay? But the surface of the quinoa seed, if you look at it under the microscope, is like a golf ball. It has lots of little holes, and because you have a sticky pericarp, it's impossible to completely remove that pericarp with the saponin without also going into the embryo and part of the perisperm and removing some of the nutritional quality of that seed. The alternative method is to use a washing method and then drying, which is using water and energy in order to be able to get rid of the saponin. These are really, really good questions because we keep going back and forth over 15-year cycles between, at least in the Andes, between breeding sweet quinoas, saponin-free quinoas, and bitter quinoas to avoid bird predation. And it's not a question that has been resolved. I think I have a potential answer in the tertiary gene pool. Non-adhering pericarp that's inflated. We see it in a number of different diploid species, but we haven't made crosses yet to try and remove that. But that would answer the question of utilization of water for and retention of nutrient content.
Thank you.
Thank you. Can you go ahead with the second question for the second candidate?
Yes, please.
So thank you very much for your presentation. My question is, do you have any ideas about the contribution of your SNPs on the phenotype variation?
Actually, there was a big phenotypic variation, as we have seen in the table. So I think but we have like phenotypic variation. But mostly the variation that we see is controlled by genetics, because we had a heritability of 0.63.
Okay. And do you think that is really enough for breeders to take into account those SNPs for their breeding programs?
After validation, we can use them in marker-assisted selection. So I think yes. Thank you.
Thank you very much. You can now go to the coffee break. Please get back at 11:30 when the next session will start. Might I ask to Kaima Ikan if here to approach me, please? Thanks. Hello, good morning everyone. Please have a seat. We are about to start this session on agricultural use of soil amendment, fertilization, and microbes association. I would like to give the floor immediately to Chaham Ikam for her presentation. And please, thank you very much.
Good morning, everyone. Can I start? Sorry? Can I start? Yes. It's okay. Good morning, everyone. I'm pleased to be with you today in this third LAFOBA forum, and I'm honored to present to you the essential of my work entitled Biostimulants Applications Improve Durum Wheat Yield and Soil Health Under Drought Stress Conditions. This work was carried out at the Laboratory of Excellence in Agrobiotechnology and Bioengineering at the Faculty of Science Mlalia Qadi'iyya University in Marrakesh and presented by me, Shema Ikan. Durum wheat is a key cereal that especially the Carioca variety, and it's belong to the family of Poaceae, with a national production of 8.1 million tons, and it's mainly grown in regions like Shawiya, Abdal, Haouz, Sedla, Gharb, and Sais. Globally, countries like Canada, United States, and those of the Mediterranean basins produce about 31.4 million tons. Durum wheat is rich in a source of complex of carbohydrates, protein, fibers, vitamin B, and also it constitutes a source of energy and contain antioxidants enzymes like lutein and carotenoids. Despite this nutritional importance of durum wheat, it faces several challenges, and this brings us to the problematic that we are facing and talking about. One of the main constraints is the continuous increase in the world population that will reach 10 billion by 2015, and this means that the need for food has increased, and which pushed farmers to use excessive chemical fertilizers to meet with this growing demand. The use of these dangerous fertilizers have also aggravated the situation. The intensification of climate change and the increase in the population is leading to the scarcity of precipitation, which is aggravating drought. In this regard, it is necessary to think of new sustainable approaches, such as the use of plant growth promoting rhizobacteria and organic amendment like compost to mitigate the negative effects of drought stress. For plant growth promoting rhizobacteria, they are characterized by the production of exopolysaccharides, production of phytohormones, nutrient acquisition, induction of antioxidant system, secretion of ACC desaminase, and also fixation of nitrogen. For compost, they have 4 roles to improving the physicochemical and microbiological properties of the soil, improve water and mineral retention capacity for the plants, increase soil porosity, strengthen plant microorganisms associations, and also improving plant tolerance to abiotic stresses. In this regard, this work aims to evaluate the efficacy of biostimulants and comparing PGPR and compost to increase the yield and tolerance of durum wheat to drought stress under greenhouse conditions, and also to investigate the comparative effects of these combined technologies and chemical fertilizers, including nitrogen, phosphorus, and potassium, on growth and biochemical mechanisms underpinning drought tolerance in durum wheat. For the biological material, it consists of compost based on green waste, PGPR consortia, about 2 strains, and also for the vegetal material, it consists of Carioca variety, durum wheat, and for chemical fertilizers, including nitrogen, potassium, and phosphorus. For the experimental material, we have used 2 water regimes. The first one, well watered, about 75% field capacity, and the second one about drought stress, about 25 field capacity. Different treatments were applied, including treatments without any biostimulants like control, and other treatments with the application of biostimulants either alone or in their combination. Moving to the results section, drought stress decreased the growth rates. However, the application of biostimulants, especially compost alone or the double combination between compost and PGPR, increased the seeds numbers, spikelet height, 100 seeds weight, and also the shoot and root dry weight, under 25% field capacity compared to control plants. As for nutritional parameters, we have recorded an increase in the phosphorus content, in the phosphorus content about 111% compared with the control plant under drought stress conditions. As for biochemical treats, drought stress hamper the activity of antioxidant system. Furthermore, the application of biostimulants, and especially PGPR, compost, or the double combination between them, especially in the activity of polyphenol oxidase, under 25% field capacity compared to the control groups. Biostimulants not only have a positive effect on the growth physiology biochemistry, but also they improve the physicochemical properties of the soil, and we have noted an increase of available phosphorus, cation exchange capacity, total organic matter, and total glomalin through the application of PGPR alone or their double combination between compost. Drought stress not only hampered the antioxidant enzymes in the plant, but also in the soil. However, the application of compost alone improved catalase activity by 50%, and for the peroxidase activity, they show higher values through the double combination of compost and PGPR in comparison with control groups by 43%. And for alkaline phosphatase, we have recorded higher values in amended plants with compost by 254% in comparison with control groups under 25% field capacity. In general, the presence of biostimulants, and especially compost and PGPR, improve biomass, physiology, biochemical traits such as the biosynthesis of osmolytes and osmoprotectants, the activity of antioxidant system, and also the production of hormones, and the improvement of physicochemical properties of the soil. As a perspective, we aim to conduct omics analysis, including metabolomic, proteomic, ionomic, phenomics, in order to identify stress-responsible gene to tolerate drought stress. And thank you for your attention.
Thank you very much. That was very much interesting. Are there questions among you that you would like to ask? Yes, please. The microphone is on the way. Thank you.
Where is the presenter? She's gone. Oh, okay, all right. My question is, the compost, it contains plenty of microbes. So how you can dissociate the effect between organic matter brought by the compost and its microbiota? And also for your PGPR, treatment, it would have been good to use exactly the consortium, which is desactivated, just to kill cells, to see whether there is any amendment that were brought by the bacteria or the media at least. So I want just to listen to your comment. What do you think about that?
Maybe we can also let the others do their questions so you can reply to all of them. So there is one from there and another one here.
Thank you very much for your nice presentation. My question is about the quantity of compost you used in the trial. You said that you used only 5 grams per pot. I think it's not sufficient, and also I guess that you may use 10% of the volume, maybe, but 5 grams, I think that it's underestimated related to the results you got. Thank you.
Yeah, just a quick comment on this PGPR business. I've been hearing about this personally. I'm getting old, and I've heard this for the last 40 years or so. What intrigues me is no one is trying to open the box to see what are we exactly talking about. So last month, Ken Giller has published a real article that I invite everyone to look at. It starts like this: science losing its way, and commenting on some of these black box approaches. Because when we have been following something for 40 years without showing hardcore evidence or science showing that a mechanism indeed enhances the cereals using soil microorganisms, I think it would be time to start opening that box. But I invite you to read the paper by Ken Giller. Thank you.
Thank you for your presentation. I have a question about how did you choose those bacterial strains? They are commercial strains, or you selected that and isolated?
Okay, so please, you can answer, otherwise they become too many. If you can remember it, or otherwise we ask it again.
Thank you for all these questions. For the first question of the professor in front of me, for the microorganisms in compost and also in PGPR, in our experiment, we have already serialized the compost. So this gap is in the perspectives. We aim to know the native microorganisms of the compost that we have used. So that's it, because we sterilize the compost and then we combine the effects between compost and PGPR to know the synergistic or the dual combination effects on wheat plants. So that's it. For the second question of Professor, about compost 5 gram, our objective is to find a low quantity that have most effects. So we have conducted not even this experiment, but several experiments on several plants to validate that we need to use low quantity because the cost is too expensive. So and also we focus on this dual or triple combination between compost, PGPR, or even EMF like arbuscular mycorrhizal fungi. So if we find a tripartite combination with low concentration, that will give us more, or that will boost the tolerance of plants, this is the gain. And for the question of Professor about PGPR strains, they are not commercialized, so we have selected these 2 strains from several strains based on the PGP traits, like the production of phytohormones, secretion of exopolysaccharides, like the tolerance to polyethylene glycol. So we have choose these strains based on several parameters. Thank you.
Thank you very much for the
too.
questions and for the answers. I was suggested to keep the questions of next presentation to the end. So I will now give the floor to the second speaker, who's Driss Touami, please. Thank you very much.
Good morning, everyone. Thank you, Julia, for sharing this session. and thanks everyone for being here for my presentation and for inviting me to present my work. So my name is Driss Touhami. I'm an assistant professor from... okay, it's good? Perfect. So my name is Driss Touhami. I'm an assistant professor at the College of Agriculture and Environmental Sciences at Team 6P, Ben Greer. So sorry, today I will not be talking about salinity per se, but I will be talking about a love story between inorganic pea and insect frass, which is a type of organic amendment, and how those two different things are combined and can help the plant to mitigate salt stress. So as you may know, with climate change problems and also with growing population coupled with different problems, we are basically not in a hospitable planet anymore, because 6 of the 9 planetary boundaries has been crossed so far. Moreover, soil, which provides 99% of our production, is treated by salinity around the globe, and this issue is spreading over more than 100 countries, and therefore it's providing from producing enough food for people, but also lower quality for the food that is produced. And more than importantly, this salinity issue is costing the world more than 30 billion annually from crop loss, but also is reducing crop and soil microbe diversity, but also having an impact on carbon sequestration. And therefore, we have to think about some solutions to increase production and sustain production in these kind of saline conditions. Also, you may know that phosphorus is very important for crop production. Plants, they need this element to do a lot of physiological processes and to increase root growth. But under saline conditions, phosphorus availability is decreased. And therefore, a lot of scholars and scientists, they've been applying phosphorus fertilization to increase plant production. But phosphorus fertilizers, the normal or the common phosphorus fertilizers used, they have low P use efficiency because there is absorption of the phosphorus into aluminum iron oxide, there is a precipitation of the P into calcium, and therefore they have this low P use efficiency. Then we have to think about some other forms that can circumvent those problems and help us to increase P use efficiency and increase nutrients P uptake. But also, the application of organic matter is very important to provide, of course, nutrients, microbes to the plants, increase soil health, but also help to improve physical properties of the soils. As you can see, insect frass now, which is a byproduct of insect farming. So insect farming is a very booming industry right now. The estimated market value is around $17 billion by 2032, while insect frass market value is estimated to $1.5 billion by 2032. So insect frass has been used as an organic amendment in different cropping systems around the world. But the knowledge gap is that we don't know how insect frass can be used to mitigate salinity. And we don't know if when we combine insect frass with different forms of phosphorus, how they interact positively or negatively to increase and mitigate salt stress in different plants, especially some plants that are very important, such as corn. So the objective of the study that I did with my master's students was to combine for the first time insect frass with different form of phosphorus to look at the both soil properties, especially P availability, but also below and above ground biomass parameters, but also looking at the functioning, especially photosynthetic activity, as well as nutrient contents. So to do so, we took a low P soil from the farm at Bingria site. We added, or we increased the salinity of this soil by adding sodium chloride, and we used 3 different sources of phosphorus, polyphosphate form and 2 orthophosphate form. So KTTP, which is a polyphosphate form, is a form of phosphorus that is inorganic, but it has PO link, and therefore the microbes, they need to degrade and break down this link to be able to be available for the plants. So we included also 3 rates of insect frass that is coming from black soldier fly. So 0, 100, 200 milligram per kilogram of insect frass. And then we had, of course, positive and negative treatment or control. We used a pot with 3 kilogram where we sow drachma as a corn variety. We grow the plant for 45 days in a randomized complete block design with 4 replicates. Going now to the results. So from the study, we found that in terms of plant growth and the biomass, of course, salinity has a detrimental effect and decrease the plant biomass and the plant growth. But we found that the application of fertilization mitigated this effect, and KTTP was the best form in terms of increasing the plant production and growth. But we also found that applying insect frass didn't have that much, positive effect we wanted. And we also saw a trend, especially for the application of F1, which was the 100 milligram per kilogram. It has a kind of negative effect. And 1 and 2 actually best combination were with KTTP when we didn't apply any insect frass, and when we applied 200 milligrams per kilograms, which increased the above ground biomass, especially shoot. So going to the root, so looking at the above ground, going now to the below ground, we found that salinity, of course, again decreased root length, surface area, root volume, but increased root diameter, which is, of course, an adaptation of the plant to limit, sodium uptake by the plant. But we found also that the P fertilization, alleviated those impacts of salinity with, again, KTTP increasing root biomass, root volume, and root surface area compared to TSP and MAP. Insect frass also, again, didn't have that very much wanted effect, but we also saw that if one had a decreasing and significant, negative impacts on root trees. In terms of plant nutrition, salinity had no significant impact on root P, but increased the shoot P, which is maybe due to a deletion effect, especially for the positive, control, where you have higher yield, and then the P content decrease because of the dilution effect. But most importantly, in terms of potassium content, both in shoots and roots, we found that KTTP really was the best to increase the potassium content in the plant. And we also observed that the application of insect frass at high level tended to increase, but the application at the lower level had a detrimental effect and decreased the plant potassium. The best combination was, again, the addition of KTTP, which is a polyphosphate, with the F2, which increased both shoots content of phosphorus and potassium. Going now to the sodium and also the ratio, potassium and sodium, which is a very important indicator of salt stress. And let's start with the sodium first. And of course, salinity will increase this ratio, content of sodium in both shoot and root. But you can see that polyphosphate was really amazing to decrease sodium content in the shoots, but also in the roots. But something very interesting again is the application of insect forage, especially at F1, increased shoot sodium content. And then I will, in the conclusion, try to discuss those kind of effects. But the striking effect again is the impact of KTTP, especially in increasing the potassium and sodium ratio. So you can see it's performing very well compared to TSP and MRP fertilizer. In terms of functioning, and especially phosphosynthetic activity and P availability, we saw that KTTP again was the best. So KTTP was my friend during this this experiment, and he gave us very good hope, and we are actually continuing to work on that. So increased both chlorophyll content, photochemical efficiency, but also the availability in the soil of phosphorus. And then we found that F2 had higher and tendency to increase both Olsen P in the soil, but also chlorophyll content and also photochemical efficiency. And again, F1 had this negative impact, and the combination of KTTP with F2 was the best combination that helped us to have more functioning and more availability of P in the soil. We did also PCR results, and those results showed that we had the first component, which explained 35% of the variability, and the second component explained 25% of the variability. But the first component, as you can see, is mainly driven by the plant nutrition components or parameters, but also some of the root and growth parameters, while the second component was mainly driven by physiological components and also some other components related to soil fertility. And as you can see in the different groups, we can see the green one on the top left, they are the positive control, while and they were like colocalized with the good shoot potassium and sodium ratio both in shoot and root, but also root length to increase nutrient acquisition and so on and so forth, while the negative control was on the other hand side, and it was mainly colocalized with sodium in the soil, but also in the shoots and root, while the KTTP was in a different group where it was mainly colocalized with different traits related to physiology, but also to plant growth and root growth. So, as I said, KTTP was the best form that can help plant to mitigate salinity, and this because of polyphosphate, as I said, is a long chain of inorganic P, and then it has this bound between P and O, and it needs to be broken down to be available for the plant. So this slow release capacity helps the plants to have a high availability of P in the soil, and then the roots can grow, as you can see in the difference between the different treatments in the picture there. So this better root morphology had a positive effect on, of course, the nutrient acquisition, especially the potassium and phosphorus, but also help the plant to have more physiological functioning and therefore biomass acquisition. in terms of insect frass, so the effects were rate dependent. So we showed a tendency with F2 to have a positive effect, affecting availability in the soil, physiological functioning, but also a little bit of increase in the root. But on the other side, which was very striking to me, and the lower rates had a negative effect. Yeah, in fact, as you saw maybe in the thing, the graph, the sodium in the shoot was really high, and then it has a detrimental effect on root growth, on shoot growth, and also functioning and photosynthetic activity. While we found the best combination, because I was working on the integrated nutrient management, so the best combination of inorganic phosphorus and insect frass was combining polyphosphate with the F2, and this combination give us higher phosphorus availability in the soil, but the plants also, they did great photosynthesis, and also they have a good ion homeostasis, where phosphorus and potassium were higher compared to sodium and compared to other orthophosphate or combination. So, take-home message: to have a better plants to grow under saline conditions, you have to choose very wisely your source, especially for inorganic fertilizer. So here we talk about inorganic P, and then polyphosphate was the best compared to orthophosphate and TSP, which are very wild, available fertilizer and commonly used by farmers. Second, the combination of insect frass and polyphosphate was a great strategy compared to only one strategy, either organic or inorganic. And therefore, it is very important to combine and to look and to work on integrated nutrient management because of this, but also to reduce the cost of fertilizer and to work on a circular economy perspective, which is very important for climate change. I would like last to thank all the collaborators worked with me, and some of them they're still working with me, especially my master's students who is in Spain now working on plants and genetics. I think he got enough with soil, so I think he moved to another thing. Thank you so much for your attention.
Thank you very much. Yes, you can stay and we keep in mind the questions for the end of the presentations. I would now like to give the floor to Sofia Baha, please. Thanks.
Hello everyone, my name is Baha Sofia, I'm a PhD student. I'm working in developing biostimulants from plants and algae to mitigate the harsh effects of the major stressors, which are drought and salinity in arid and semi-arid regions. But here I will present an essay which was done to evaluate the efficacy of Piganum harmala seeds and Moringa oleifera leaves and their potential as plant biostimulants on crop performance under saline conditions. Okay. So I guess we already know the global context. We have the booming increase in global population. Thank you. The booming increase in global population and the important decrease in the arable land, and we know the continuous severe change in the climate, so and its direct impacts in the food security, and the numbers are scary. We can see here that 700 million people faced hunger in 2023, and about 2 billion people faced moderate to severe food security in 2023. So, because our topic today is about salinity, so around 30% of the measure of the world irrigated land is already impacted by salinity. This is globally, and for Morocco especially, we have that around 700,000 hectares of land in Morocco are affected by salinity, which 500,000 of it are in irrigated areas. So why were we scared of salinity? Because salinity causes the oxidative stress and osmotic stress in the plants and the ion toxicity, which can imbalance the nutrients and cause nutrient deficiency, and which has direct effects on the growth of the plant, its delay, its germination, and lower the productive yield, its quality and quantity. Okay. So I have some economic numbers. To highlight the importance of the salinity, because we know salinity affects the plants and affects the agriculture and limits its productivity. Agriculture productivity is limited, so the economy is affected. So we have heard that more than 27 billion of global annual agriculture are lost due to salinity, because more than or about 50% of the crop can be lost due to saline sensitivity of the crops. And salinity affects already, we have mentioned already, that around 30% of irrigated land globally. Also, the scientists, the experts, the professors and researchers have invented and developed and also adopted some technologies and sustainable practices to mitigate and to limit the effects of the salinity that threaten the agricultural productivity. We have soil management, water management like desalination, crop selection and crop breeding, whether we have integrated farm management, some like precision agriculture and models for resilient agriculture, and some biological approaches like soil amendments, biochar additions, biostimulants applications, which is our hero today. So what is biostimulant? Biostimulant is usually defined by its function, not its composition, because the source of biostimulants can largely differ. So biostimulant is any substance or microorganism that can improve the plant's growth, either by improving its nutrient use efficiency, improving its resilience and tolerance to abiotic stresses like salinity and the drought, and that's it. So the source of biostimulants or the type of biostimulants can largely differ. We have mentioned, we have like, the main of them are flavic and humic acid, protein hydrolysate and amino acids, ketosane and other biopolymers, seaweed extracts, plant extracts, and other beneficial elements, and also the microbial biostimulants, as my colleague has mentioned before, like PGPR or rhizobia and mycorrhiza microorganism. So the economic market of biostimulants is under development, is in going development, and for 2025, this year, the market was estimated that have reached more than $4 billion United States dollars, and it is projected to reach more than $7 billion United States dollars by 2030. But actually, the biostimulant market is underdeveloped in our continent, Africa. So for my essay, the methodology is simple. We have two plant materials as a source of our biostimulants. We have done an aqueous extraction, water-based extraction, and alkaline-assisted extraction. And then we have done a phytochemical screening for the bioactive compounds that can have the biostimulant effects from our extras. Then for our crop, we have used the barley, the steppe crop that is largely cultivated in this area. And we have tested different concentrations from 0.5 to 20 milligram per mils, and we have done a germination test under normal conditions and under saline conditions, and we have assessed the germination percentage and the vigor index. So we have seen here that the composition of extracts can largely differ just based on the source, and also the extraction method can impact the composition of our extract. So here that moringa from water extracts had highest yield and the highest DPPH activities and the highest tannin content, while the same plants at the alkaline extract was the richest in bioactive compounds and showed the strongest antioxidant activities. The Piganum harmala seeds already has not showed like a moderate bioactivity. So the take-home message was that the extraction method can strongly shape the composition and the efficacy of the biostimulants, because in the next slide you have seen that the response of the seed to the extracts can differ based on the source, the concentration, and the extraction method. So here for the saline conditions, Moringa olivera leaves, water-based extracts had showed the significant outperforming outperforming controls and other treatments in germination speed index, and also for the alkaline extracts showed the longest early development of the shoots, while Piganum harmala water-based extracts at 20 milligram ml concentration had produced or had influenced the length of the roots, surpassing both glycine betaine, which is an amino acid that have been chosen here as a control positive, and also had surpassed the negative control, which is soaking with the water. For the saline conditions, the results differ also based on the conditions, if they are normal and saline. For the germination speed index, normally Piganum harmala has influenced both germination speed index and root length, dispersing both controls, negative and positives. The same for the shoot lengths. So, as a conclusion, that this result can confirm that the type of extract, the plant source, the stress conditions all alone can influence our results. So that's it.
Thank you very much. Please, you can stay here for the questions that will follow the last speaker, which I invite now, who is Saeed Korki, please. Thank you very much. And also the first speaker who talked, you are invited as well here. Even if you already received your question, you can participate to the rest of the meeting here. Thank you very much. Thanks.
Thank
Can you stay between 10 and 15 minutes?
Yeah, it's okay. I will try. I'll try my best. Yeah. So, hello everyone. Thank you so much. I would like to thank the organizing committee for this amazing conference. Thanks a lot. So, my name is Saeed Khorshi. My presentation today titled Mining Hollow Torrent Microbes from Desert Ecosystem as a Promising Solution to Enhance Cell Stress Tolerance. Where is this? How can go? Sorry. How can tweet back? So, I'm not going actually to talk a lot on the salinity, on the effect on crop, the crop response, etc. I think you received already a lot of things relating on the effect of salinity on crops. So I would just want to mention that also Morocco is facing this abiotic constraint with almost 500,000 hectares located in the irrigated area in Morocco are influenced by the salinity, and which of course cause a significant losses in terms of crop productivity. And in this picture you saw here, you can see is due to the irrigation, the accumulation of salt. This is actually in the Zaguras region. So as this issue is not recent, so a lot of approaches were suggested to alleviate or to enhance the salt tolerance. So among these approaches, there is use of phytoprotectant, there is agronomic cultural practices, also breeding programs, biotechnology solution, etc. But today we are going to focus on this aspect of biofertilizers. And this actually biofertilizers application, including bacteria, fungi, microalgae, etc., they show the great potential to alleviate the negative stress of salinity. And there is quite several mechanisms, including regulating the osmotic balance, reducing the reactive oxygen species generation, including the scavenging process. Also, resuming the plant growth by producing the growth regulators. So in this project, the objective is to isolate and identify halo-tolerant microbial strains from desert ecosystem. With this ecosystem, they have contrasting salinity gradient. The objective also is to characterize and isolate the halo-tolerant strains for multiple plant growth promoting trait, and also to investigate, to try to relate this functional trait to the original niche of different microbial isolates. And also at the end to design the halo-tolerant microbial consortia and evaluate their effectiveness in alleviating the salinity stress in durum wheat. So in this project, we are working on a different ecosystem in the southern of Morocco. And you can see here in this map, there is really different ecosystem, including river, lagoon, sabha, dune, grara. So those are really different ecosystems that are specific to this region of Morocco, and they have great differences in terms of soil, of physical-chemical properties, the nature and the type of vegetation, and also for sure the microbiome. And in this presentation, I'm going to focus on these three ecosystems, including dune, lagoon, and grara. So let's start by dune. So dune, as you might know, is we call it in Moroccan Darija rug. It's a sand landscape known by its limited vegetation. It is actually, we have actually sampled this in our way to Asari. On your right hand, you can see these dunes. So we sampled some plant there, and we try to isolate microbes from those plants. We also sampled another ecosystem called dune. So what is a dune? So a dune is a kind of saltwater intrusion. So it accumulates the water of the sea, since it is near to the sea. And also, I'm also working on grara ecosystem. So this grara ecosystem, I'm actually focused on there's another PhD student who work especially in this ecosystem, and he will give a presentation tomorrow. So in this ecosystem, I just want to try to compare, the species that are found in the ecosystem to the other two main ecosystem, which is dune and lagoon. So in terms of plant species, so I'm working, we actually sampled 4 plant species in the dune ecosystem and another 5 from the lagoon ecosystem. And you can look at the differences already. So it's totally different, the type of vegetation from an ecosystem to another. I'm also focusing on this tree that I found in the grara, which is Vacholia tortoris from the Fabaceae family. So for this sampling, we've sampled 3 types of vegetation, let's see. There is tree, shrubs, and small vegetation. And from this, we take both rhizosphere and bog soil, the shoot and the root, where we will isolate our microbes using serial dilution techniques. And then from this, we will screen them for their ability to tolerate the salt stress. And then for sure, we purify our halo-tolerant microbes. And then we will functionally screen them, for different plant growth promoting traits, as you may see here first for drought tolerance, phytohormone production, including auxin, ammonia production, phosphate solubilization, etc., etc. And then from this in vitro screening, we're going to design our consortium as follows. So our strategy first is based on the contrasting salinity. So we will choose different isolates with different ability to tolerate salt stress. And then we will consider what we call here the inter-ecosystem consortia, meaning the microbial isolate that compose the consortia are coming from different ecosystems. And of course, we will try also the intra-ecosystem consortia, meaning picking up the isolate from the same ecosystem and try to look at their efficiency. Also, in terms of plant species, as we know, this microbiome and the type of microbial isolate and also their function can vary among the plant species. So we're going to use also this approach to consider the interplant species consortia and the intra-plant species consortia. And of course, is to end up with with an efficient halo-tolerant consortia with multiple PGP trees. So those consortia to be tested under greenhouse conditions, with the objective is to evaluate their effectiveness to mitigate salt stress and also investigate the physiological and molecular mechanisms by which this isolate may alleviate the salt stress. So we will really look at the physiology of the plant and the response to different microbial consortia. So if we're looking at the results, so the three ecosystems are actually totally different in terms of physical-chemical properties. If we look here, for example, at the soil pH, so we are dealing with ecosystems that are totally alkaline, as you can see here, even though there is some few differences, but still alkaline. Also, if we look at the electrical conductivity, so there is a gradient of salinity, actually with the lagoon ecosystem being the most saline one compared to Grara and Dune. And also, if we look at the soil fertility, so we can see that Grara has more organic matter, and also in terms of micronutrients such as nitrogen, phosphorus, and potassium, you can see that Grara has always the high levels of soil fertility, I mean in terms of micronutrient content. Also, if we look at the species, I mean, the Grara looks like the most fertile ecosystem compared to Dune and Lagoon, which is, I mean, looks logical, as Dune is only sandy soil and Lagoon is a kind of saline ecosystem, so it's generally halophyte plant. So in terms of diversity and distribution of isolated microbes from this ecosystem, so we get around 31 isolate, halotolerant bacterial isolate. They are distributed as follow. As you can see, 50% of this isolate belongs to the bacillus, and it's actually our spore-forming bacteria, which is normal since we are working on extreme environment. So this kind of isolate are really adapted to this such extreme environment. If we look also at this in the distribution of this microbial isolate among the three ecosystems, you can see the really differences. So for sure, the most common one is the bacillus. As you can see, it is common between the three ecosystems. But if we look, for example, to the Dune ecosystem, we can find Pseudomonas is really specific to this Dune ecosystem. Same for Grara. The Grara is actually the most diverse ecosystem. As you see, it contains different genera of bacteria. And also, if we look at the Lagoon ecosystem, it's more Vegibacillus, and this Metabacillus is really specific to this ecosystem. So from those results, we can already see that each ecosystem has a specific microbiome, has a specific halotolerant bacteria isolate. So when combining this, it can really, I mean, the different isolates may have different mechanisms in alleviating this abiotic constraint, as this ecosystem normally are, let's say, our extreme environment. So we did also the salt tolerance. So first we screen our bacteria isolates based on a basic level of salt tolerance, which is 5% salt. And then also we subject them to the higher, let's say, increase in salt concentration. And if you see here, if you look at the lagoon is we seen is the highest saline ecosystem, and it actually hosts the highest number of halotolerant bacteria isolate, which makes sense. I mean, also if we look at the lagoon, which is the second one after the Dune which is the second one after lagoon, it makes sense also because the Dune is just here, so they are near to the sea. Again, we look also at the drought stress. As you know, the salinity and drought are always connected as a biotic stress. It's almost the same response, as you can see. So the thing is, there is significant variation in terms of drought tolerance among the different ecosystems that you can see here. And also, the bacterial drought tolerance vary not only between ecosystems, but also between plant species, as you can see. The plant species might host, for example, if we look at the first plant species here in the Dune, the isolate H2 showed lower tolerance to drought than the two other isolates. So there is really huge significant variation in the ecosystem and also in the plant species. Also, the Guerrera ecosystem being the ecosystem that hosts the highest number of drought tolerant, which is, again makes sense, as this ecosystem is a dry ecosystem, as you can see from the picture that I showed before. And here we try actually to link this abiotic stress tolerance of isolated bacterial isolate to their native niche. And we notice that if you see here, the highest salt tolerance bacteria are actually belongs to the lagoon ecosystem, which is a saline one. And again, to the dune ecosystem that we see here. So that means that this bacterial isolate tend to retain their ancestral function. And this function might help actually in designing this microbial consortia to alleviate the salt stress, in crops. The same thing we noted for drought tolerance. So for drought tolerance, you see the Guerrera is the driest ecosystem. And actually the Guerrera contains the highest number of drought-tolerant bacteria, which makes sense, again, lead us to the conclusion that these bacteria isolate tend to retain their ancestral function. And also, if you look at the ER production, which is, we see the same trend following the abiotic stresses. So there is huge significant between the three ecosystem, where of course we can see here that the bacterial isolate with high production of ER, we can see them in the dune and lagoon ecosystem, and this can link to the salinity, and we know the role of auxins in alleviating salt tolerance. This is very interesting as a treat. So I'll go a little bit faster since the time is not here. So for phosphate solubilization, we know this is the same thing. If you see the isolate in the Guerrera ecosystem, they didn't show significant differences. They coming from the same plant, but didn't show any significant differences in terms of phosphate solubilization. Again, if you look at the lagoon also, the same species can host the different bacteria isolate with different capacities to solubilize phosphate. And coming to the nitrogen fixation also, what we notice is dune and Guerrera ecosystem host the highest number of nitrogen fixing bacteria. And this actually can be explained with the presence of Fabaceae family plant species. Over there, if you look at the Growtheria irrigators of Fabaceae, this may be host more, highest number of nitrogen fixing bacteria. Also, this tree plant, Vachuolia totalis, is also a legume, belongs to the legume family, so this might explain this highest number of nitrogen fixing bacteria. So I'm now going on this. This is an ongoing experiment where we test different halo-tolerant microbial consortia. So for the conclusions, for sure, the three ecosystems exhibited significant differences in terms of soil physical-chemical properties, but in terms also of microbial diversity. Also, the halo-tolerant bacteria was strongly associated with saline ecosystem, which is more lagoon. And also, these bacteria tended to retain their adaptive trait to their original niche, which makes this ecosystem actually as a promising target to isolate halo-tolerant microbes, of course, for developing efficient halo-tolerant microbial consortia. And in this work, I would like to thank really the team that are contributing to the to this project. I'd like to thank Professor Hijri, Professor Harish, and Professor Bobo for their guidance. Also, I'd like to thank Phosboucraa Foundation for funding this project, for this amazing collaboration. Thank you for your attention. Sorry, I didn't do this.
Thank you very much to all of you. Actually, the time of this session is just ended, but if we are allowed, maybe one question. Is it okay? Okay. Yes, please be quick, because we should be already starting with the next session. Thank you very much. And thank you all.
Actually, can you hear me? So actually, it's not a question, it's a comment for Professor Touhemi. I think it's really interesting, and I was triggered with the data you showed with polyphosphate and compared with other form phosphate. From my perspective, it's really to make sense, because for phosphate transporters, there are two categories. One first categories, it's proton-dependent, H+ dependent phosphate transporters, and second one are sodium-dependent phosphate transporters. So when you are giving a lot of phosphate or available phosphate as DSP to the plant, so it's H+ dependent transporter that they are activated. While when the phosphate is deficient, so it's mainly an A+, sodium dependent phosphate. And that makes absolutely sense that polyphosphate is not immediately available. So the plant, they are trying now to get the energy from sodium to increase or to uptake phosphate. So in terms of mechanistics and phosphate transporters, that makes sense that polyphosphate are far away more efficient than other form of phosphate.
Thank you very much. I think we leave the floor to the other session. Is it a super quick question that you have?
I have just a question for Mr. Said. Comment or question. So why you didn't complement your work with the metagenomic approach, just to separate between cultural effect or contribution of those consortia and what is non-cultural, which is a very considered fraction?
You have it there.
I think it's okay. Thank you so much for this comment. I didn't mention this. We are actually under processing this. We are going to do metagenomics to look at this different soil, root, shoot, box or rhizosphere soil collected in those ecosystems. And we are going again to go for metagenomics in our greenhouse experiment to make sure that our microbial consortia are persistent in this soil. Thank you so much for your question.
Okay, so there is time for last one. And do you mind joining us here so that everyone can hear you with the microphone? Because I don't know where it is. Oh, no, it's just over there. Okay, it's on the way.
Thank you very much, Said. So my question is actually, in the soil, in the greenhouse, are you using the same soil that you had from the fields, or are you trying these consortia in a different type of soils?
Thank you so much, Sikhay, for this question. So the answer is no. We are not using the same soil. We are actually trying to use different type of substrate. So we are using now a substrate that is totally artificial, and then we will move to the soil, to the naturally saline soil. We're actually going to launch the greenhouse experiment here in Laayoune, as I'm working in Asari. So we will be launching the greenhouses and the field experiment also using a naturally saline soil. Thank you so much.
Thank you very much to all of you. I'm sorry we have no time for other questions now. Maybe we can continue during the lunch break. And I give the floor to you now for next session. Thank you very much, and sorry for stealing a few minutes.
Thank you so much. So we start a new session, and with our keynote speakers who cannot join us personally, but I think he will join us virtually. So our Professor Herbert from KAUST in Saudi Arabia. I will ask the people connecting. Oh, here he is. Okay. Sorry for not being with us. So please introduce yourself and give us your presentation. I think you have around 20 to 25 minutes. Thank you. Go ahead. We don't hear you so far. Could you? Not yet. The microphone is not working. We cannot hear you so far. Could you start? We have some technical problem. I think it will be resolved in a few seconds. Okay. Here we are. Okay. Could we postpone your presentation once we resolve the problem of the microphone? Now it's working? Not yet. We don't. It's your microphone, I think, who doesn't work. Take your...
now?
Oh, now, yes. Yeah, much better.
Okay. So
thank you.
It wasn't working beforehand,
Oh,
so
much better.
I'm very sorry for all these issues about technical things. But now you see me, but you don't see my presentation.
Yes, we see you and we hear you, so you can start, please. Yes.
Yeah. Okay, so basically I'm working at KAUST, which you probably know is in Saudi Arabia. And I've been working there since more than 10 years. We started a project actually to go into the desert and look how plants can actually live and survive there under the extreme conditions of desert conditions. So I'll try to show my presentation now. You see that now? You see my presentation? I hope so. So basically we try to analyze soils and plants and microbes under these conditions. And so this is, I'll give you basically just like an introduction. Here, the soil foods system. Of course, we have plants, we have soil, we have a lot of organisms that live in the soil, and everything has to work together. So if one of these components is actually not working or not present, you have a problem. So that means, of course, in the desert, when you don't have vegetation, you have a serious problem, because the vegetation is really feeding the whole system in the soil, and the soil is feeding the plant again. So then there's a problem. We have a number of projects that are running in the lab. So one is a desert microbiome project, where we have isolated many microbes from the desert plants, and then we try to use these in agriculture and in regreening. We have a mangrove project that tries to also use microbiomes and for restoration in deserts, but also in agriculture. I will touch two projects. One is a native genome project, where we characterize ecosystems for functional desert restoration. And one is the soil bioatlas project that actually looks into the properties of soil. So the soil atlas project is a project where we analyze the physical, chemical, and the biological properties. We've been doing that at 92 specific sites, the representative sites in Saudi Arabia, to get an overview of how different soils are and how different properties they have. And then, so I will just show you how that works. So here we have a satellite mapping. This is actually the NEOM region where we do regreening. And then we have different regions that we are mapping that are in the regreening projects. Here's the team that goes out then, and then of course we do the soil analysis that goes back into the lab. And here in the lab, we are actually doing different levels of soil analysis. One is of course a physical soil analysis that's very simple, very classical. But then we also do here more sophisticated analysis, ICPOS, which where you can basically do all the elemental analysis. And we also have XRD, X-ray diffraction, to really come down to the crystal structure on the soil chemistry. So this is a more classical way to analyze soils, but then we are putting in another biological assay. So we can look here by metagenome sequencing, the 16S for bacteria, 18S for protists, ITS for fungi, and different other combinations of primers for looking even into animals and plants that lived there in this area. So we can actually restructure complete landscapes, even there is no organism living there anymore, but the DNA is left in the soil, and so we can actually say what lived there hundreds of years ago or thousands of years ago in the soil. So you can do that in terms of archaeology or of course also in agriculture. So it's a great tool that I think we developed there. And then of course we isolated also rhizobia and mycorrhiza, which are very important components, as you know, for plant functioning and for soil functioning. And all of this together is like coming into our microbial biobank. So this contains like more than 10,000 different microbial species that are all functionally characterized in terms of genomics and functionality. And so here we also then did a large-scale project with different other colleagues in the institute to characterize different ecosystems in Saudi Arabia. And so here I give you just a small film about this project, which basically shows you also a little bit about the diversity of landscapes and of ecosystems that you find in Saudi Arabia. And of course, then what we do here is to try to catch all this biodiversity by really high omics methods, genomics, microbiology, metabolomics, transcriptomics, so all the tool set that is available. And then we are going out and we are collecting samples and then bring them back and analyze them in the laboratory. And here you see that Saudi Arabia is really having a rich biodiversity of ecosystems that one can look at, that one can study. And so there's thousands of indigenous species that exist in different areas. Yeah, and so all of this is actually then going into a genomic database that will be publicly available. So that will be a reference system for desert research. And really then, of course, the idea is to make this available to other researchers so that systems such as this can be really analyzed in depth. Some of the plants that we have been analyzing in these systems, so these are small annual plants, perennial plants, shrubs, trees, and also some of the marine marine plants that are also included. Yeah, so then what do we do with all of this? We basically put that together into a package for a strategy development system. So here we have basically an AI-based method to look into a plant system and a soil system and say, okay, what is the best plant and what is the best microorganisms that are actually needed to restructure this ecosystem to have a sustainable ecosystem. And this is actually done by a startup company that we created. So people can come and basically we give do an analysis and then give a report and what is the best strategy for planting there or for doing agriculture and things like that. Just here some issues about how to make crops fit for climate change. So I will give you two examples of the work that we did on heat and drought resilience of crops. So using microbes coming from desert plants and then trying them out in crops. We have been doing that extensively in different places. For example, here in ICBA in Dubai for the, so it's a very experimental site. We've been doing field trials on wheat, barley, and alfalfa with different strains that we isolated from these desert plants. And as you can see, this sometimes has really big success because here, for example, this is one of our best strains that we have been isolating for making plants resistant against the salt, but also heat tolerance. And so over different seasons, you can really get enhanced biomass, but most importantly, you get also increased yield up to 50% sometimes. So it's quite a great tool, and it's just a single microbe that we actually spot on the seeds. So it's seed coating technology that's behind that. And when we looked into the mechanism, because we found that this is like a heat stress, which also works in the lab. So we went back and tested how this works. And of course, to find the mechanism, we then go back to Arabidopsis, on which it also works nicely. And in Arabidopsis, we found that this is actually linking to what is known already, to what we call thermopriming. So here, basically, temperature that you can add is changing the chromosome at certain positions. And these positions are actually the positions of the heat stress genes. So what happens then is that if you are giving this microbe, the microbe interacts with the plant, and the plant actually gives information that leads to metabolic reprogramming of the microbial side. And we identified this component, which is a small sulfur metabolite that then in the plant is converted into ethylene, and ethylene is necessary for this epigenetic priming event, and this actually makes plants heat stress tolerant. Here is another example for Pseudomonas for water stress, for drought stress efficiency. And we tested that also on a number of plants in the field and in the lab. And this is also can make plants really nicely drought tolerant. So again, we went and looked what happened in Arabidopsis for the mechanism. You can see that, for example, when you do Arabidopsis growth on 25% PEG, which is a kind of a mimic of drought stress, these plants don't like that at all. You can see they're really suffering. But if you have been colonized by this one microbial strain, SA190, they are really growing much, much better. So what is happening? We realized that actually the plant is having much better water status. It does not stop growth as the wild type, the non-colonized plants do, which are basically going and freezing in and not growing anymore. And what we found is that actually the key probably is in the water use efficiency. So these plants have much higher ability to use the little water that is left in the soil, and thereby it gives them a huge advantage. It makes them very tolerant also to that, and we've figured out that some of the metabolic reprogramming on the microbial side here, we still haven't figured out what the molecule is, but we know that on the plant side, this is again an epigenetic priming mechanism that is in this case not depend on ethylene, but on ABA, which is of course a very important hormone. Yeah, so one thing more, what can we do with these mangrove bacteria? We can actually use these guys because mangroves, you know, are growing in high salt water, and they have their feet also in the water, which plants do not like. So we tried actually on different plants again. And here I just show you a few slides on that we identified where the plants are growing much better on rice. And so we can increase the productivity of rice grains. So as you can see here, single strains or double strains, so dual incubation gives you up to 50% more yield. And one very important thing that we found is that actually the life cycle of the rice is shortened massively by these plants. So the productivity is increased, but the life cycle is changed by 1/3. So that means these plants can actually produce in 2 months and not instead of 3 months, which could already solve, I think, the major problem of feeding the world, because 1/3 of the population of the world is feeding on rice. How about on salt? I mean, salt, when we have these 2 microbes, you can see we get in number of grains per plant, a number of weight, about 200–300% increase in the yield. And here is the rice that we can get. So it's really massive potential here for agriculture. Finally, if I still have some time, I will just tell you something about microbes that are living in the seed. So here we did a study with an orphan crop, which is called fonio. So this is a millet grown in West Africa. It's one of the oldest indigenous cereals in West Africa. That you might know, can grow in really nutrient-poor soils and drought tolerant, and it's largely undomesticated. That means you have problems in lodging, you have problems in seed shattering. And so here, as you can also see, that this is a study here we did with Simon Kruttinger at KAUST. Fonio seeds are really tiny compared to rice or wheat or maize seeds. And what is also very evident, if you look at this scheme here, the yield is really low compared to the other crops, and it's actually not changing because it's actually not developed. So we were looking into these geoclimatic factors, whether they are shaping the fonio genomic population structure from a microbial side. But Simon was actually looking at it from the plant side, which most people are, of course, are starting to do. So this is an interesting crop because it really grows in many different regions across West Africa. And these regions are really characterized by either high salt levels, high altitude levels, very little water levels. And the structure of the genome, of the fonio genome, is really also structured according to the geoclimatic factors. As you can see here, for example, you have temperature, latitude, longitude, altitude, and precipitation. They're all impinging on the different genetic factors. But we were interested in whether these plants are also containing different microbes that are transmitted now by the seed, and then that the seed already carries different properties, and they're not coming only from the genomic side of the plant, but also from the genomic side of the microbes. And just a few slides here, it just shows you when we take now, these are different varieties of fonio from different areas, and you can see these are different microbial compositions that are very different. And of course, then the question is, we used 223 different varieties and tested whether they have different properties. And indeed, you can see, for example, the soil, the pH, the structure of the soil, the precipitation, all of that have actually an effect on the microbiome composition that is carried over in these different seed varieties. So we then also did an experiment that is, I think, quite interesting, is that we took 3 different varieties which have different seed microbiomes, And then we were growing them on a soil that is very rich, so it's a nice greenhouse soil. And then we looked into the next seed generation. And you see that some of the microbes, actually most of them, are kind of maintained in the next generation of the seed, but some are lost, and some others are actually gained from the soil. So there is a real dynamics possibility, partial loss of microbial communities, but also a new integration of new microbes. So the plant really selects what kind of microbes it doesn't only want to do live in one generation, but in many generations. So I think that's really important for doing thinking about different varieties. And we also did GWAS with these different fonio, and we found a number of gene loci that are connected either to development of the seed or of the embryo, and of course many of them also together with immunity. Yeah, so basically I think what I want to say is that a host genome is really connected not only to its climate, but also to it, and not only to its geographic location, but it's also connected to the seed microbiome, which of course responds to climate, to soil, geographical localization. So it's the holobiome theory is that there's a co-evolution of the host and the seed microbiome. And last not least, I think I've finished with my presentation, is the people that I'm working with. Here's my group and my collaborators. And I think I stop here and take some questions. Thank you very much for your attention.
Thank you. Thank you so much. Thank you, Dr. Hobbs. So, any questions for Dr. Hurd? No questions? People that work on selections of... Okay, go ahead, please.
Okay. Can you hear me?
Yes.
Yeah, thank you very much for your presentation. Impressive piece of the data. So my question is regarding fonio microbial communities. Any evidence or any clues about the fungal communities associated with fonio, and particularly for mycorrhizae that you have talked about? Thank you.
Yes, of course, very important. And unfortunately, we did not do, we didn't go beyond the 16S and the bacterial population. We just wanted to actually do that in the first case study, because when we started, it was not clear at all whether there is any connection really between the seed microbiome and the environment. And I think this is, but we're doing this now with other species. And we included also the different fungal analysis and a much broader analysis that we can do. So clearly, I think, you know, all different microbes are involved. It's not just bacteria. This is like just the top of the iceberg.
Thank you. Thank you so much. Any other questions? So, okay, go ahead. We need to translate, I think. Could anybody could, okay.
السلام عليكم ورحمة الله تعالى وبركاته. سؤالي: ما هي أكثر الطرق فعالية لتحسين إنتاجية الأراضي الملحية في المناطق الجافة، مع الأخذ في الاعتبار تأثير الملوحة على نمو النباتات وخصوبة التربة؟ وشكرا.
I don't know if did you get the translation? Not yet?
No.
No. I think the question is, what are the best way to increase the productivity and the selling conditions based on your experience in using those technology?
It's okay. This is a very good question. I think the first thing to do is actually to do a real thorough soil analysis. So I think if you don't know what the conditions are in your soil and what functions are missing, what microbial functions are missing in the soil, you will have a very poor success rate on we do a lot of regreening now, but also I think the same is true for growing any type of plant. So this information actually gives us the necessary input, what we have to bring back into the soil, how to make a degraded soil or a poor soil into a fertile soil.
Thank you. Thank you so much. So I think with this, we can go ahead and go and ask our PhD student. And the next speaker, it's Hafsa Debagh. Please come in. Present yourself.
Good morning, everyone. Thank you for attending my presentation. I'm Hafsa Debagh, PhD student here at Asari. And today I'll be sharing with you insights from my research on microbial soil properties and microbial communities, as well as their interaction. Okay. Can we change the slides? Okay. Here? No, can you show me? Hello. Okay. Okay. So desert ecosystems shelter a large diversity in terms of flora, which is facing many threats, including drought, salinity, high temperatures, and overgrazing. But despite all these conditions, these plants manage to adapt thanks to adaptive mechanisms they have developed. They include morphological, physiological, and biochemical. These plants play an essential role in ecosystem stability, fertility and structure, and the overall balance. And in recent years, in recent years, focus has turned to root-associated microbiome for their key role they play in nutrient cycling, stress resilience, and overall plant health. In my study, I focused on 4 plant species, Lavandula coronopifolia, Lycium intricatum, Nitraria retusa, and Cercia tripatita. This choice was based on ethnobotanical studies that were conducted among another project. Okay, so for objectives, the first one is to assess rhizospheric soils from different locations in Laayoune Saqia al-Hamra region. And second, to study the variation of microbial communities between species through the analysis of root-related microbiome. And third, to understand the ecological mechanism related to the interaction between soil and microbiome. All these objectives will allow us to better understand the ecological mechanism of every plant species. The map shows the geographical distribution of our study sites. And for sampling, we targeted 4 sites for each species, and in each site we collected 5 replicates of rhizospheric soils and roots, which were immediately stored under appropriate conditions for further analysis. Soil samples were analyzed for a range of biochemical analyses, including electrical conductivity, pH, available phosphorus, carbon content, and elemental composition. For roots, from roots we extracted DNA and performed sequencing using a 16S rRNA gene sequencing, and then the data analysis. So moving on to results. We can see that for organic carbon and inorganic carbon, the Cercia tripartita and Erytusa showed similar results, and they were also Lycium intricatum that were significantly higher than L. coronopifolia. For the electrical conductivity, we can say that Erytusa shows the highest levels. And this is in accordance with the nature of their habitats. And for pH, Lavandula showed the highest levels, followed by Lycium, Nitraria, and finally Cercia. So this PCA biplot shows three clusters. We can say that Lavandula coronapifolia is more associated with metal-rich environments, and whereas lithium intricatum is more present in alkaline soils and also rich in magnesium. And for CR satipartita, it was highly correlated with high electrical conductivity and organic carbon. Now moving on to the relative abundance of microbial communities. There are similarities among species and between species, with the Pseudomonadota and Actinomycetota presenting more than 50% of total root-associated bacteria. But also there are also some similarities between and within species and sites. For the alpha diversity, we use three indices, Shannon and Simpson. The Shannon index showed significant differences between within Lavandula and CR satipartita across different sites, and no differences for the two other species. And the Simpson index only showed significant differences for Lavandula coronapifolia. The beta diversity, we used the Permanova and the principal coordinate analysis. Well, the principal coordinate analysis showed significant differences between sites for all the species. And the Permanova showed that a total of 42.5, 35.9, 30.5, and 48.8 of the variation is explained by geographical sites for Lavandula, Lithium, Nitraria, and Cercaria, respectively. And finally, we have in the redundancy analysis. For Lavandula coronapifolia, we can say that the potassium, pH, magnesium, and calcium contributed the most to this variation or composition of microbial community. For lithium intricatum, we have phosphorus and magnesium, and for the rest of the species, we have a broader range of soil parameters. And in conclusion, we can say that species distribution reflects soil quality preferences. The relative abundance of taxa in root shifts depends on geographical sites. Phylum level composition show little variation, but genus and species may reveal clearer differences. And finally, we can conclude that each species shows distinct and soil and microbial responses. Therefore, a specific species-specific approach is essential to understand this interaction. That would be all for me. Thank you for attention.
Thank you so much. Yeah, you can sit down here. Yeah. Thank you so much, Hafsa, for your good presentation. So, any questions for Hafsa? Go ahead. Please, excuse me. Your microphone? Okay, she's coming. Huh?
Okay, very interesting. Congratulations. Very nice piece of data. So you have really a very strong variation between your site, Lavandula, and with the others. So this is a really very, very important. It's very rare that you have such big differences between these sites. Do you know why?
You can come in here.
So I'm speaking about, alpha diversity.
Hafsa.
This can be explained by the nature of the ecotypes. Maybe the first site was different than the three others. For example, we're having Phosboucraa versus Sagradas. So maybe this can be related to the type of ecotypes.
This is the type or geographical location?
No, it's the geographical location. Yeah.
Any other questions?
Thank you for the presentation. I have only one question about, thank you to showing the importance of microbial inoculation
as an alternative to improve plant resistance to silent stress. But what the aim after the characterization of your microbiota, if you can use it after as inoculum, if what? Yeah. Okay.
Well,
and
okay, finish.
yeah, and the second question, they are only a characterization about bacteria. I want to know if they are the same thing for mycorrhiza, for example, and if yes, how you can do to prepare your inoculum and use it to enhance plant resistance to silent stress. Thank you.
Okay, so for now we just worked on bacteria. We're not planning to work on any other thing like rhizopsia, as you mentioned. So now the focus is only bacteria. Maybe later within other projects we can see other aspects.
Okay,
yeah.
and the aim is to use these strands to inoculate maybe other plants? Or
no. In fact, for now, this is not our objective. We're just exploring what's the communities that are related to each species. Okay. And this work may be a basis for future researchers.
Okay.
Yeah.
Thank you so much.
Thank you. Okay, another one over there, please. Excuse me.
السلام عليكم ورحمة الله. معك الخليفي سيد أحمد، مزارع من منطقة الجريفية بوجدور. بما أنك درتي البحث على هذه المناطق الصحراوية كاملة، بغينا الخاصية اللي تمتاز بها إقليم بوجدور، لأني شفت في البحث عندك درتي إطلال على المناطق كاملة. شكرا.
شكرا على السؤال. هو في الأصل حنايا ما شفناش كل إقليم بحد ذاته. حنايا الهدف تاعنا كان هو ندرسوا كل اسبيس، كل نبته، ونشوفوا اشنو هو الاختلافات اللي كاينين ما بين المناطق، ولكن بلا ما ندخلوا في بزاف تاع لي ديتاي.
Any other question before we go to the next speaker? Thank you, Hafsa. Could you applaud Hafsa? So now our next speaker is Anas. Could you come here and start presenting your research work?
Thank you. Thank you, Professor Adwan. Hello, everyone. Thank you for being here. I'm Anas Bourza, PhD student with Professor Abdelaziz Hirish, and I'm working on rehabilitation of phosphate mining land with OCP's group. Today, I will share with you part of our finding on how blue panicum can help to rehabilitate phosphate mining land using organic amendments. But before diving into the result, let's talk about phosphate mining. As you know, phosphate mining is vital for agriculture, but it leaves behind a degraded soil. For that, OCP's group is actively working to reverse this damage through sustainable restoration. Among species was tested on restoration, but the blue panicum has shown potential, but we lack sufficient data on its performance on this type of soil. Among the species tested on restoration, blue panicum is until now the best. And this is the main goal of our research. Now, let's understand why restoring myelin land matters and what challenges we face. Restoring myelin land it's part of OCP's plan to protect nature and create opportunities for the farmer, but myelin land are poor in nutrients and don't let plants grow easily. To solve these problems, our research aims to test blue panicum performance in tailing mining soil and how different amendments can help to improve biomass production and soil condition. For that, we conduct an experiment in a controlled greenhouse in Ben Guerir. We use pots of 3 liters and a plant density of 200,000 plants per hectare to be similar in the field condition. We use two types of soil, Saline mineral soil from sedition and phosphate seed, and agricultural soil from a nearby plot. And also, we use two types of organic amendment. We have compost with 20 tons per hectare, and also manure with 40 tons per hectare. We used a randomized block design with 6 treatments and 5 replicates. We have Saline mineral soil like a control, Saline mineral soil with compost and also with manure, agricultural soil, agricultural with compost, and agricultural soil with manure. Now let's pass to the result. As you see, plant height of blue panicum showed a highly significant difference across treatment. The application of organic amendment to the Saline mineral soil markedly improved plant height ratio level comparable to those observed in agricultural soil. But for stomatal conductance, they didn't show any significant difference between the treatments. Like you see, for biomass production, you can see in the pictures there is a highly significant difference between the treatment, especially the Saline with other treatments. Until now, we have the highest biomass production in Saline mineral soil with manure with 7 tons per hectare per harvest. Let's pass to see the percentage of mineral content. As you can see here, there is a huge difference between the part of the content of mineral in shoot and root, and each element we will see it alone in the next slide. Let's start with the nitrogen. Nitrogen content showed no significant difference between the treatment into both shoot and root. The concentration of nitrogen was notably higher in leaves compared to the root. For the potassium in shoot showed no significant difference across the treatment. Root potassium was significantly higher in all treatments except agricultural soil, indicating adaptative nutrient uptake in mining soil. For the phosphorus showed a highly significant difference in shoot and root across the treatment. Root had higher concentration than shoot, especially in mining soil, which is naturally rich in phosphorus, leading to strong phosphorus accumulation in root under this condition. The calcium didn't show any significant difference both in shoot and root between the treatment. Also for the sodium showed a very highly significant difference in the root. And now let's pass to see the heavy metals cadmium. As you can see in the graph, there is a highly significant difference in both shoot and the root across the treatment. If you can see, we have the concentration of cadmium in height in the root under all treatment with mineral soil, indicating strong retention at the root level. This ratio can explain that, and organic amendment on haze cadmium retention in the root with the highest ratio observed in the Saline mineral soil with manure and also with compost treatment. The lowest ratio in the Saline mineral soil indicates greater cadmium mobility and translocation to the shoot in the absence of amendment. And this correlation matrix showed us that cadmium has a negative a negative correlation with biomass and also with the height of plant. Like a conclusion, organic amendments improve biomass production and help regulate nutrient and soil dynamic in phosphate mining soil. Blue panicum with organic input demonstrates a capacity of cadmium photostabilization, storing heavy metals in the roots and minimizing their translocation to the shoot. Integrating organic amendments is key to restoring soil health, enhancing plant productivity, and reducing environmental risk. Thank you for your attention.
Thank you so much. Thank you for your presentation. Any question for our PhD student? Okay.
Thank you. Two questions. I will start with the most difficult one. This is really something I was really surprised that the cadmium is negatively correlated with biomass. This is something really weird and strange. And I would just to understand. Normally, plant cannot discriminate between cadmium and calcium, right? And normally, it's really well documented that cadmium is really biomass dependent. So my question is, did you measure the biomass of roots and shoots and then the proportion of the cadmium that is stored in?
Thank you for this question. Actually, yes, when we have cadmium high level in the shoot, Test. If you can return to our graph, please.
Go back.
Just go back.
Yeah, you can
No.
Did you go back to the presentation?
Okay, there is no problem.
Go back now.
Yes, this for the control. We have a high level in cadmium both in shoot and root. As you can see here, both in shoot and root, and for tailing mineral soil and tailing mineral soil with compost or with manure, the concentration of cadmium is more in the root. But lower in the leaves.
So the biomass fresh weight or dry weight?
The biomass dry weight. Can you repeat, please?
No. So it's dry biomass, right?
Yes.
Okay. So my second question is a very, very quick one. Why didn't compare it other plants for revegetation? Why are you using only blue panicum? I would expect testing a triplex or other shrubs that are more perennial or some trees. Why blue panicum?
It's a perennial crop, like Dr. Redwan said, but this is just part.
It's a perennial grass.
But that is just a part of our what we do. We have also a lot of crop like carobs, like fig, like also we have cisbania. There is a lot of, but this is just a part of our finding.
I think there is
another question
over there. Okay, go ahead.
السلام عليكم ورحمة الله تعالى وبركاته. أولا أعبر عن قلقي وحزني الشديد للأضرار التي يعني الأضرار الناتجة عن المناجم على التربة. لدي سؤالين. السؤال الأول: ما هي الآثار السلبية للأنشطة المنجمية على التربة والنباتات في المناطق المحيطة بالمناجم؟ وكيف يمكن تنفيذ استراتيجيات فعالة لاصطلاح الأراضي المتضررة وإعادة تأهيلها بيئياً؟ السؤال الثاني: كيف تؤثر عمليات التعدين على خصوبة التربة وتركيبتها الكيميائية، وما هي الإجراءات التي يمكن اتخاذها لتقليل تلوث التربة بالمعادن الثقيلة والمواد الكيميائية الضارة الناتجة عن الأنشطة المنجمية. وشكرا.
السلام عليكم، شكرا على هذا السؤال. دابا لو سي بي الان كتحاول انها تستغل الاراضي اللي كانت ديجا استغلتها من قبل، تعاود تحييها. وما بين الحوايج اللي انها كدير هي انها كل عام عندها اوبجيكتيف ديال 1000 تقريبا 1000 كتار اللي خصها تريابيليتا. والشطر الثاني صراحه الله من السؤال ما عقلتش عليه، الا كان ممكن تعاوديه.
السؤال الثاني او الاول؟ السؤال الاول او الثاني؟
ثاني.
نعم. كيف تؤثر عمليات التعدين على خصوبة التربة وتركيبتها الكيميائية، وما هي الإجراءات التي يمكن اتخاذها لتقليل تلوث التربة بالمعادن الثقيلة والمواد الكيميائية الضارة الناتجة عن الأنشطة المنجمية؟ وشكرا.
بالنسبه للسؤال صراحه له استعمال ديال كما كيقولوا الغبار، استعمال ديال الغبار كينقص من هاد الدائره هذه. وكيف ما شفتي هنايا كنقدروا نرجعوا الارض بحالها بحال اي ارض عاديه، يعني الغبار بواحد لي دوز معينين كيعطيونا دي ريزولطا بحالهم بحال الاراضي الخصبه. شكرا.
Thank you, Sianas, for this nice presentation. So my question regarding the cadmium accumulation or translocation to the plant. Really, it's a kind of surprising that this blue panicum plant accumulates more cadmium in the root than in the shoot. Do you have an idea what is the mechanism behind this? I mean, the mechanism that the plant accumulates only cadmium in the root and the shoot. Is there, absence of some transporter that takes the cadmium? Because generally, in the case of the crop, it's normally the inverse. You will find more cadmium in the shoot than in the root. Do you have an idea about this? Thank you.
And this is the goal what we have in this research, that we find that blue panicum stock cadmium in the root and not in the shoot.
I think it's not only for cadmium but also for sodium. If you see that this crop accumulates more sodium, which means that it stops the translocation of the element from the root. I would say not root, but rhizomes, because it's a rhizome, okay? Then it allows we have less translocation, and we find that in the sodium. And one of the things we've been growing the blue panicum on the farm in Fumilwent for now over 6 years, and we didn't find that much salinization of the soil. And the major reason, I think, it was that the sodium was more accumulated in the rhizome of the plant. Okay? Other questions? Go back. Ladies first, then I come back to you.
Thank you so much, Anas, for your presentation. My question is regarding the water. We know that blue panicum has shown good results regarding the salt stress, but I think that it needs water, a lot of water. So do you have any idea about the crop water requirements during a cycle production? And did you use any sensors to measure the amount of water you were giving for irrigation? And thank you.
First, I want just to clarify the difference, what the advantages of blue panicum. As you know all, for alfalfa, they need, I think, 12,000 meter cube per hectare. But for the blue panicum, it's just 600 ton per hectare. Okay, it's the half. You asked me also about No, I didn't use it.
Okay, please. It's working? No.
Yeah, it's on, it's on.
Okay, it's working now.
It's working. I want just to comment on the question. بالسيدة اللي طرحت السؤال بالعربية، يعني أنا العربية ديالي مش مزيانة، غادي نحاول نرد بالإنجليزية. But it's really very important to understand, I mean, how the OCP and make the operations. So we are talking about different rock phosphate. So this is sedimentary rock phosphate. And the way to process it and to collect this rock phosphate is to remove the sterile part. So there is some layers that do not connote phosphate. And what they do, it's actually just to remove that part, what we call it topsoil. They put it aside. And then once they reach the layers of phosphate, then they collect it. So it's really not really a real tail as processed in other mining operations. And those that contain trace element or heavy metals are naturally occurring in soil. So it's not the problem of the metals are themselves. The problem is their bioavailability. If they are taken by plants or if they are taken by animals, that's the problem. And to do that, they have to be bioavailable. So they need water and to be the uptake. But metals, heavy metals, are absolutely everywhere. Okay? So, of course, this may enhance erosion. And that's why the technologies of revegetations are coming, developed to stabilize not only soil from wind erosion, but also to avoid leachate of trace element. But we are really in arid environment, so this is really challenging as it is. But in temperate areas, revegetations is a technology that has developed since decades, and they are used. And if I give the examples of Canada, that country that I know much better, they have really issues because they grind the rocks and they extract metals, and then this is a real tails that is mixed with other chemical elements. But for sedimentary rock phosphate, there is sterile, remains sterile as natural soil. That's it.
Thank you. Thank you so much. So I think, last question. Okay, go ahead. Please be quick because we have to go to lunch.
Thank you. Quick question. Thank you, CNS, for your presentation. I just wanted to know, since I'm working too on cadmium in the soil, I just wanted to know if you did study the impact of your crop on cadmium complexation in your soil. Does it help in cadmium, does it differ, or did you compare the impact of your crop compared to other crop? Does it change anything regarding cadmium complexation, of course, in correlation with the different physical chemical parameters of your soil? If yes, then how did it change in cadmium complexation in your soil? Did it affect cadmium absorption, etc.? Thank you.
I don't really understand your question. Can you repeat it, please?
Okay, so does your crop impact cadmium in the soil? Or did you manage to do any type of analysis regarding cadmium in the soil? But I'm not talking cadmium in plants, but in the soil.
For the moment, I don't do any analysis until now.
Sorry?
I didn't do any analysis from
the soil. No. Okay, thank you.
Okay, so I think with this, we please applaud our speakers. And we invite you now to go directly to the lunch. The door is just over there. So please, and we'll see you in the next session, I think, after 1 hour. Thank you. at UK Centre for Ecology and Hydrology in the UK. He is actually chairman of the working group on water, food, energy nexus in ICID and a chairman of the British National Committee on Irrigation and Drainage. He's the founder of the working group on the use of non-conventional water resource for irrigation. He's got hundreds and hundreds of publications on water management and also on modeling. He reviewed more than 20 international journals. One of the most important things that Dr. Ragab made is developed a widely used model, what we call SALT model, for field scale as a tool for integrated management of water, soil, crop, and nitrogen fertilizer. So it's an integrated hydrological model system for catchment basin scale. So he will be with us to give us a keynote speech. So let him to come in and present his presentation. And he's got 25 minutes, but with the time, hope we can get it shorter so we can manage the next speakers. Thank you, Dr. Ragab. Come in.
Thank you, Dr. Shukrallah, for this generous introduction. Good afternoon, all. I would like to start by thanking Dr. Abdulaziz Harishi and the organizing committee for inviting me to give this presentation. My presentation today will be really focusing on new technology we used to accurately estimate the irrigation water requirement. My focus will be on the irrigation side only today. And I would like to start by talking about what are the challenges that we have. And as you see here, that by 2050, we need to increase our food production by almost 70 to 100%. And also, we have to take into consideration the water use is expected to increase by 50%, and the increase in global energy will also increase by 30 to 40%. And there is currently a gap between the water supply and demand, and also the climate change actually is not making it that easy for agricultural production. I will show you why actually we have to care about not using too much water for irrigation. This is an example of our work in Cyprus, where you can see there is a gap between future supply and the demand. You can see here the demand is going up and the supply is going down, and this is the gap. And this is in Cyprus. Now, we did similar work also in Italy, and whatever the scenario used in 2 catchments in Italy, this is a work of a PhD student. And also the gap in Italy also is widening, and that gap is increasing over time. In Brazil, I had also 2 postdocs who came from Brazil, and they have been also working on climate change and the impact on the Brazilian northeast catchments in the area of Recife and Fortaleza, if you know Brazil. And also it shows that there will be a reduction in the recharge to aquifer, and also reduction in surface water to the rivers. Our work also in the UK, although UK is a humid country, but also we are facing drought events and more frequently, and also we are facing the same prediction that there will be a gap between future water supply and the demand also in the UK, although it's not a dry country. So it is a worldwide phenomena, and we've got to be really prepared for it. So why really we have to care about crop water requirements, and we should only add the right amount? And because if you add more water, especially if it's brackish water, you are also adding more salt. And also, if you add too much water, you leach the nutrient, not only the salt, but the fertilizers and the nutrients. And also, you decrease the soil and groundwater quality. You decrease the water productivity and water use efficiency. And also, you are irrigating less area than you should. And also, you're wasting a lot of water, energy, and labor. So the crop water requirements, the way actually we estimate it or we measure it, it's either by equations, and that could be meteorological equations or empirical equations, and also by soil measurements like soil moisture deficit, and also plant measurements like sap flow. You can also use lysimeter to estimate the crop water requirement, and also you can have direct or indirect measurements like the ones that I will show you in a minute. So for the definition of these different crop water requirements require that we have to know the ET node. We refer to the reference evapotranspiration from well-watered short plant like alfalfa or grass. And when we multiply that by a crop coefficient, we get the crop evapotranspiration, or what we call it ETC. But however, ETC could be actual and also could be potential. So we've got to be careful between the two. Potential, it means you have available water, and when the actual is that when you don't really have the maximum water availability in the soil. So how we started to measure the evapotranspiration and the crop water requirements, we started, of course, by the simple thing, Class A pan. And then Class A pan, of course, it's no biological control in the evaporation, and people started to find a correction factor. They called it Class A pan. And then came the technology of weather stations, and meteorologists found it is now time for them to develop equations. And we have seen so many equations appeared in the literature, and all of them really either based on temperature or radiation or both. And that was a breakthrough. However, they were really classified either based on energy or temperature or a combination. But one equation in particular took the plant into account. It was the Penman-Monteith equation. All the other equations, they didn't really take the plant into consideration. Only when Monteith in particular came to work with Penman, these are two British colleagues or men, they have been looking at what actually the role of the plant in the evapotranspiration calculations. So they came with this equation, and that's the Rolls-Royce equation of evapotranspiration until now. However, a lot of people couldn't find it easy to apply because that plant representation parameter, which is the RS, that actually made that equation not to be easy to a lot of people, especially the extension services and so on. So the ICARDA and the FAO, they started to work an approximation for that canopy resistance value. So they have been coming with a different approach to reduce the Penman-Monteith equation to manageable equation that everyone can use. And if you look at the canopy resistance or stomata resistance, actually it fluctuates during the day and during the night, and also from the beginning of the season to the end of the season. So what actually has been done, they assumed that if we take an average seasonal value, then we can simplify the equation. So this is really what they did. They assumed seasonal value of stomata resistance or canopy resistance or surface resistance, 70 seconds per meter, and they applied that into the original Penman-Monteith equation, and they arrived at this actually equation that now you don't see anymore, that RS. However, that has been, of course, investigated by so many people and to try to see the validity of this equation. What we did, we had a project funded by European Commission, and we had a site in north of Italy in Bologna area. And in that site, we have been using the edicovariance and scintillometer and also Cosmos technology, which I'll elaborate more. So that's the site where we have here the site of the instruments located. And then we started to use the edicovariance. The edicovariance is, as you see here, you have those sensors here. They can measure the flux. And that flux here, it could be the evaporation flux, could be the CO2, and could be methane as well. We use it for methane at the same time. So it depends on the sensors, really, that you add to the edicovariance instruments. And so it would actually measure the turbulent flow that goes here between those sensors, which is horizontal flow in the direction of the wind. And that actually gives you an idea about the flow or evaporation flux. That's one instrument we used. And then we used another one, which is the scintillometer. And scintillometer it is a transmitter and the receiver at the end. And the idea of the transmitter is the same as edicovariance. And you send here infrared or microwave from one end, and you receive it at the other end. And actually, that beam here is, you have that current circular flow affected by the temperature and the humidity, which is caused by the evaporation from the different vegetation. And that can actually measure you the actual evaporation over a length of up to 10 kilometers. We tried that in Crete for 6 kilometers. We tried that in Zimbabwe, and also we tried that in India. And then the measurements that we had on using scintillometer edicovariance, using also the wind direction, we could have the footprint of the evaporation, and we can see where actually most evaporation happens between the transmitter and the receiver. We can analyze this data, and we can, if you have a mosaic of crops together, you can still, through the calculations of software, that you can see also how much evaporation coming from each vegetation, how much coming from the wheat, like 44%, and the maize contributing 35% of the total evaporation over the transect of the scintillometer. Now, when we go to the comparison between what we measure by the edicovariance or by the scintillometer, and we compare that by the FAO modified Penman-Montes, then we find really quite a huge difference. And here, if you look at the ET node, this is the ET node, the continuous gray curve. And then if you multiply that by the crop coefficient of FAO, then you obtain these actually yellow dots. So this is the ETC calculated by the modified Penman-Montes. And this is quite high compared to the blue and the orange dots, which obtained by the edicovariance and scintillometers. And in some cases, we found the ratio is, as you see here on the top, the ratio here, actual evaporation by scintillometer compared to the ATC of the Penman-Montes modified equation is about 35%. So basically, the Penman-Montes equation, the modified version by FAO, actually overestimates the actual evapotranspiration by the crop. And that's very important. So the calculation here is giving us an idea that we have to be very careful with the way that we calculate our evapotranspiration. And one of the things that if you carefully look at, most of the equations developed by meteorologists And these equations are not developed by agronomists. And the meteorologists, they give you the atmospheric demand for water, not the crop demand. Agronomists, although they believe a lot in meteorologists, but meteorologists actually, they are by nature using the meteorological data. But agronomists would have to be careful here that there is a difference between atmospheric demand for water and the crop demand for water. What if you have any equation based solely only on climatic data, you are here talking about atmospheric demand, not the crop demand. And that's really a very important point I would like to make. Most of meteorological data that they don't have representation of the plant actually calculating the atmospheric demand, not the crop demand. And that's an important point. And also these equations that shows us that some researchers reported they can obtain the same yield when they irrigate with 50% of AT. And some they say, well, we got the same yield, 50 or 75, as much as 100%. It's because 75% of potential value, a ratio 50% even. Someone sent me a paper, he got the same yield, irrigating with 50%, exactly like 100%. 50% of something high, something big is also big. The exaggeration on the calculation of ET is actually makes some people to believe they are applying deficit irrigation. In reality, it's not deficit irrigation. It is the actual amount. But because your ET calculated as a potential value, not actual value, that's what you get. So we've got to be careful with the way we define deficit irrigation. And also some people reported a higher KC. Of course, if you have a high ATP, or potential evapotranspiration, if you divide that by ET naught to get the KC, then it's a high KC as well. So we have here to be very careful with these two parameters. So the issue of deficit irrigation, I have been writing that down on a paper recently. I was asked as a president of the International Commission on Irrigation and Drainage to write something about this issue. So I put that issue and other 9 issues, like the confusion about water productivity and the water use efficiency, and also the use of word sustainability. So there are 10 issues in that paper which was requested by the International Commission to publish to clarify what each term means. So I invite you really to read that paper. There are some actually findings I found as well. There are in India some experiments also supported that the Penman-Monteith equation overestimates. And also there is here Jordan Valley experiments using lysimeter. And also they found that there is eddy covariance 36% of what they calculate. And also another one in Portugal also found the same. So, and you will find a lot in the literature that the Penman-Monteith modified equation overestimates the crop water requirement, and basically you add too much water, and that's not really what we aim at. There are some publications here as well in connection with those. And then another subject here is the cosmos, soil moisture measurements. Why we need soil moisture? Soil moisture is important for the runoff, for the infiltration, for recharge, for evaporation, availability of water, and available water resources. That's why we are interested in soil moisture. And the way we started, I myself started, soil cores. That's everyone starts by soil cores. And then from the soil cores here, we started to go to neutron probe. And then the Environment Agency in the UK said, no, you are not allowed to have a radioactive source to go to the field with. It's not good for you, for your health, it's not good for the livestock. We are not allowed to have a neutron probe in the UK. So they started to have something without radioactive source. So they went to profile probe by delta T in Cambridge area. And then from that, we started to use that at a point of point measurements. But that wasn't good enough when you have remote sensing. So we started to use transects here, ERT, electric resistivity tomography. And that was 32 meters each transect. You can increase it to 64 and multiplication of that. And then that was still limited. Then we started to look at something else. And that's what I will come to with the Cosmos. So that's really the ERT graphs, that what you can get when you use ERT. And you can see here the change in soil moisture under the surface. And we did that also in experiments in the field when we have a drought. So we can also monitor the drought using that ERT system. And also we use that to look at the efficiency of soil moisture distribution under drip irrigation as well in the project. So you can see how the water is distributed under the partial root drying method or normal drip irrigation. Then we came to the Cosmos. And the Cosmos is started by German scientist, Victor Hess. He discovered there is ionization in the air. He took hot air balloon, went up, and he started to measure the ionization. And it was a sunny day. And he said, it could be the sun causing ionization. Okay, let us try again in a day that was overcast. There is no sun. And I will see if the sun is causing ionization in the air or not. So he took the hot air balloon and went up several kilometers, and they still found there is ionization. And that was the idea of the Cosmos. He decided there is a radiation coming not out of the sun, but this is from the galaxy, outer space. Radiation with high energy is called Cosmos. Then the story didn't really stop there, but that has been taken by my former boss, Professor Jim Shuttleworth. He was working in the UK and then went to Arizona. He started to work, how can I make this to be a device? And that's how it was developed in Arizona, that method afterwards. And then it came to that form of really tubes. And these tubes can look at the neutrons created by the Cosmos. If you have been using the neutron probe, it is the same principle. The Cosmos creates neutrons closer to the soil surface, and these neutrons, they have the same atomic weight as hydrogen. And when they collide with H2O, then they slow down, and then those tubes here, they actually count the number of slowed down neutrons as indication of how much water in the soil. So that's how it works. So we started to look at that, and we discovered that it is actually can measure soil moisture in a radius 300 to 700 meters. So it gives us area-based measurements in contrast to the point measurements by neutron probe and other methods. So that's the beauty of the method, that you can have an integrated value for area 300 to 700 radius. And then we started to apply that in the whole UK. And now we have over 55 stations across the UK. And also we started to do some stations in Asia, especially in India and China. And recently we had also Ghana and other African countries to work with Cosmos stations. And you have here the website. You can also see the website of UKCH if you put Cosmos there, and you will find the information and the data. And here you can have a look at any site, and it will give you all the information that you need about the soil moisture. So the Cosmos calibration is another thing that we have to think about it. And we have to for the first time that I used it, we had to do a calibration. So you do calibration, and we have been using profile probes at different depths, at different distance as well, and also soil cores, soil sensors, and we use salt med. So this is really the calibration point here, where we put here, that's the Cosmos point here, and we put here up to 100 meters away to measure the soil moisture in these circles. And also the biomass or the vegetation has some water, and we have also to deduct that amount of water. So we had to take a sample, but you cannot take samples every day. So we had to use salt med model, which I developed, to calculate the biomass on a daily basis, and then we can make a correction to the soil moisture by the Cosmos. And this is the salt med model, which I am going to give training on a Friday about that model to some of the PhD students. And then we started to, and that's also publications about the salt med model, if really you are interested to know more about the model. And this is really what you get as an end product of the Cosmos, the soil moisture after correction for the biomass moisture. And then you can, and that's really what we are after. What we are after is this figure in particular. So you can here have the soil moisture after being corrected for biomass, and this is the rainfall, and you can see how actually sensitive the Cosmos to any change in the rainfall. You can see already the change. And most important really is that you can use the soil moisture to calculate the soil moisture deficit, and if it's red, you don't need to irrigate. If it's red, you need to irrigate, sorry. If it's green, you don't need. If it's amber, it means mild stress. And also here you can see the comparison between the Cosmos soil moisture and in situ measurements of the profile probe, sensor and coil soil cores. And also you can see here the root mean square error is quite small, which is quite a good thing about the Cosmos. So the calculation of the Cosmos is really giving us a good value. The calibration showed us the Cosmos measurements can sense more than 100 meters away, and also it sensed up to 60 centimeters below the surface in irrigated area. 60 centimeters, about 80% of the roots of crops actually they lie in that area. So it can be used to calculate the soil moisture deficit for irrigation. And also that can save us a lot of water. This is the publications. And when this was published, there was a press release by Wiley about this important and the new technology. And also, I would like to acknowledge the co-authors of the work. But also someone has been asking about the soil moisture and the remote sensing. I'll just briefly mention. We got actually remote sensing images to calculate soil moisture, but none of them is direct. So we have really to find a way to make it direct. And also the soil moisture, what you get from remote sensing is for the top 10 to 12 centimeter, depends on the C band or L band, whatever the band of remote sensing. So it doesn't give you a direct soil moisture measurements, and it doesn't give you the soil moisture of the root zone either. I will, for the sake of time, what we did, when we get from the remote sensing the soil moisture of the top 10 to 12 centimeters, I developed a model. It's called the 2 layers model that was published in Journal of Hydrology in 1995. And in that model, from the knowledge of the top layer here, which is obtained by the remote sensing, I can calculate actually the soil moisture of the root zone. So that was really something that we had to do, because there is no way that you know the soil moisture of the root zone from only remote sensing of the top 10 centimeter without having to involve it in a 2 layers model, which works like 2 hats inside each other. And the idea is actually coming from the atmospheric boundary layers modeling. And I tried to adapt that to the soil area. So why we care about crop water requirements? Of course, we care about saving because we need to count every drop because simply every drop counts. Thank you.
Thank you. Thank you very much for a very interesting presentation, Dr. Ragab. And for the sake of time, any questions? Maybe one questions. Next. At the end. Yeah, please. Yeah, you sit down over there. So is our friend, I think Suad Al-Hajjaji, is she here? Yeah? Is she here? No? Please come in for your presentation because we are late and we leave the discussion to the end. Are you? Please.
Good evening, everyone. I'm delighted to be here with you today to present my topic entitled Using AI for Optimizing Irrigation in Salt-Affected and Arid Lands, presented by myself, Mbarka Al-Mu'attin. In dry and salty in dry and salty region farming face three big problems: water scarcity, soil salinization, and climate variability. Laayoune is a clear example for poor soil, lighter fresh water. Sadly, traditional irrigation make things worse. But now, AI offers smart solution. It helps management irrigation in real time using accurate data. Our system will be based in three main parts: communication layer, sonars in the field, measure soil moisture, salinity, and temperature. After that, the gateway collects the data in processing layer and send it for remote server. AI analyzing and data gives smart irrigation advices. And finally, the application of layer. The recommendations are sent for an application like Mark Farm to right time to saving water and improving results. After the context, let's look at main goal of project. First, we collect information on soil moisture salinity and whether using sensing and data is a base for smart decision. Secondly, machine learning helps us to predict when and how much water crop needed. Thirdly, Mark Farm is a digital tool that lets farmers manage irrigation in real time, easily and effectively. Fourth, the goal is to protect water, resources, and improve soil health. Now, let's look at the methodology we can use to reach our goals. First step: smart irrigation technology based on different fields sonars. Each type of sonar has its own benefits depending on the soil and environmental conditions. We have dielectric soils. Dielectric sonars. It's measuring electrical properties. They give precise of continuous data. We have also tensiometric sensors. Her objective to easier for plants to absorb water or not, to help to know is a plant to under stress or not. We have also ultrasonic and optical sonars. This is advanced sonars. Don't need to touch if we don't need to touch soil. The image shows three main types of sonars, each designed for different conditions and needs the field. Accept capacitive and dielectric sonars. At the figure A, we have capacitive sonars. They measure how well soil stores electrical charge, which tells how much water is in it. Figure B and C show for FDR and TDR sonars. This send electrical waves into the soil to analyze how they react. Those sonars are fast and accurate to work well for automated master tracking. The second is tensiometric sonars. It has a ceramic cap. It has a ceramic cap, Weatherfeld tube, and vacuum gauge. It measures how tightly water is held to the soil. Very useful to know when the plants start to feel water stress. And the final is ultrasonic and optical sonars. We have for the ultrasonic sonars use sound waves, and for the optical fiber sonars, of course, we use the light. Now this slide shows the real example for the hardware to use automatic irrigation using AI sonar. We see a soil master sonars. It check how yet soil is. The signal goes to the driver, which simplify the stabilize it, and the Arduino is the brain of the system. It reads the data and compares it with a set limit and decides if irrigation is needed or not. If the water is needed, the Arduino turn off relay. And the system also includes buzzer. That's her role to alert alert if we have some wrongs and sometimes goes wrong. And at LDC screen that shows master level and system status of real time. The slide shows how the smart irrigation systems works using real-time sonars data in cloud-based analysis. First, the system collects data for several sonars, especially soil master sonars. Then every 10 minutes, we do just control. After 1 hour, it begin to 1 hour. System calculates the crop water requirements. If it's enough water in the soil, the pump stay off. But if not, the turn on pump on. For the Beyond Master, one of the most crucial parameters assessing soil health is pH, because we have two type of pH, glasses electrode sonars, the most traditional, and we have also ion selective field effective transistor. We are currently working to the development of this by using application, but we are create application of irrigation. This application will integrate informations and details about plants, their degree of temperature, etc. This application will name it Morocco Farm, calculate meteo for region Laayoune, irrigation, insects, etc. This is the application, and this is just the website who explain what's in the application. For conclusion, artificial intelligence can truly transform agriculture in dry and salt affected area in the sustainable way. And thank you for your attention.
Thank you, Saad, for your quick presentation. Now, our last presentation is with Professor Abdurrasoor Al-Omran, who's professor in soil and water science at King Saudi University. He's got a master from UC Davis in water science, a PhD in soil science in Oregon State. He's the editor-chief of the Journal of Saudi Society of Agriculture. He published over 140 papers and 6 books. The floor is yours.
Assalamu alaikum. Thank you, Mr. Chairman, Dr. Radwan. Today, I'm going to present a work on my main topics that I'm really interested in. It's irrigation water conservation. And one of these methods that we use to conserve water is adding or altering the sandy soils to add biochar. That will be at the end of my speech. But I will start from the verses of Quran. وَجَعَلْنَا مِنَ الْمَاءِ كُلِّ شَيْءٍ حَيٍّ. We made from water every living things. So water is very important even when everywhere, I'm sure that, and especially in our region, that it is, we have a really harsh environment if we talk about the water or the soils or the weather. These three components will make it very hard really to grow a plant, but we're trying the best to have some agriculture activities in this kind of environment as in the Laayoune area. Let me start with most of our soils, it says here probably, is calcareous sandy soils, and with a high calcium carbonate, low fertility and high pore size sandy soils, low water holding capacity and high infiltration rate, low water use efficiency. In addition, we have also the major thing is a salinity problem that could limit the productivity of these soils. And as I said, in the hot condition. When I started at, research on how can come with the materials or soil amendments that will help and mitigate of some of these problems that I mentioned previously in the previous slide, we thought about the soil amendments. What should we do on that? And all these questions that listed here, we investigated that during the last 30 years and even more. What kind of soil amendments should be added? What is the cost of these materials? And you don't want also to have a very costly soil amendments. What types of soils? One of the critical things is the particle size of these materials that you add to the soils. Very important in sandy soils. How about the rate? What is the rate you add of these materials to the soils? And the longevity of this material. Does it stay for a long time, or you have to add the soil amendments every year or every 2 years? So all these questions are important. And where do you add it? And the effective of these materials, how is these materials effective with the saline water? And its effect also on nutrients uptake. All these points were investigated, as I mentioned before, but one of the earlier paper that I published in American Society of Soil Science is on, on the use of gel forming conditioner. That was in 1987 we published that. As you can see, this is the control, and these are the rate of the materials that we added. And you can see that with the higher rate, we are closing to the control soils. So it means that the rate that you added, it might be you are not benefit anything from the higher rate of the soil amendments that you add. So you have to really take care of what is the rate that it is really appropriate for a certain soil that you're gonna implement, or you're gonna add the soil amendments to it in order to save water. This is another one on polymers, and again it is the same almost. We tested that in intermittent evaporation and also with their effectiveness with days, whether it will stay after so many days, their effective is okay or not. And then we switch to what we have in our region. That's a clay deposit in some areas. So we went and dig for clay deposit in some area which has, smectite or high percentage of clay, and we added to the soil, as you can see in the farm or in the greenhouses. Some of the characteristics of these clay deposits, as you might see, the most important clay percentage, and the EC is, if we look for the very low EC for the clay in order to use it, and with the clay percentage too. Some other work on a clay deposit in the field with the saline, this regular water and the saline water in the greenhouse. This is how to apply the soil amendments of clay deposits. These are some of the pictures of the same work, but I will go faster on this. Now we came to the latest one, which is the biochar, and this is the title of the presentation. The use of biochar, which is lately in the last 10 years, it was promoted by so many people on using the biochar. And this is the use of biochar, an effective way to conserve water. This is very important. And biochar contains 80% of the carbon, and it improves soil properties, and all these enhance soil erosion and aeration and infiltration and so on. Biochar boasts water conservation. Definitely we
This is all the paper published by others in the literature. And our contribution in this time, our research has to, our team take up the challenge to conserve water and improve water quality of sandy soils in the kingdom. We've done so many work on that, and some already published work on the same subject. But this biochar, as you can see here, some of the pictures from my hometown, from Al-Hassa, and this is what they do. But I discovered that, it's this procedure was done many years ago with the traditional farmers. This is how they do the biochar. They collect the leaves and then they cover it with soils, and then they burn it, or the pyrolysis as with the low oxygen. And this is what we do in the lab. It's the same thing, and in the field, how we obtain the biochar. We did, as you can see from the slide here, so many experiments on soil columns in the lab, and this is, we test the pyrolysis temperature was at 300, 500, 700, and the size of the particles of the biochar itself, and where do you put it also. We tested that in the columns. We went to the greenhouses experiments. As you can see, that's the rate different with the different rate of biochars and the different growth of the plants. These are of some of the result of latest studies on infiltration and evaporation of some of the materials that you can see. It's lowering the infiltration of the sandy soils. Also, effect of quality, water quality on biochar. As I mentioned earlier, with polymers, the saline water, the effectiveness of polymers drop very drastically. Whether biochar, while using biochar, this drop was not been, noticed. So you can use also biochar with the saline water. And this is the experiment that we published only, one paper from this work, and we are in the way to publish the second paper, which I am presenting today. This is how we prepare the biochar, and this is the layout of the experiment. I have to go faster, okay? All right. And this is the layout of the latest experiment on the use of biochar with one of my students. He's from Yemen. And this is how we add the biochar in the greenhouses and in the open field with the fresh water and with the saline water too. This is the characteristic of biochar and soils. And roughly, because this is, I would like to mention here, the EC of biochar is very critical for you have to be careful which materials that you choose, because some of them are, with high temperature, the EC is rising up. So you have to be very careful to use the biochar sometimes in your field. These are the result of the experiment, as I mentioned, with regular water and with the saline water, and with the 100% of ETC or 80%, or this is the deficit irrigation that we use in the experiment. And these are the fresh weight and dry weight of a plant, and you can see that there is at biochar at 80% we have the better result here with the fresh water, and also with the saline water at 80%. With biochar, we have a good yield. So we save water, and we have a good production both in saline water and in fresh water. These are some of the results too. This is the figures on interaction effect between salinity and deficit irrigation, and the use of biochar. And these are the transpiration and photosynthesis. All these parameters were measured by the student who worked really hard on this. And interaction effect with saline and non-saline on total yield. And this is, I mentioned earlier, at 80% we have a good yield, and we have to stop. Okay. Radwan telling me that you have one to stop. I will just go briefly on the conclusions. This is the conclusion. The adding biochar at 5% improve the tomato growth, and definitely at 8% of ETC we have a good result. Also using the saline or definitely fresh water gave a better result, but saline water, which is reasonable prediction with using the biochar. Thank you very much for your time and listening.
Have a seat. So, session for some questions. Anybody? For Dr. Ragab, for our PhD student, or for Professor? Yes, please.
Regarding the Cosmos sensor, have you been trying in private orchards? It's available for sale, or it's still in experimenting way, or what is the current situation?
Come in here.
Please. The Cosmos instrument is commercially available, and it has been really used also for orchards. If you are asking about orchards in particular, with German colleagues, and also there was a big project in Spain using the Cosmos, and yeah, it's commercially available.
And if it's not confidential, any idea of the price?
I think if you are looking for one tube with all the trimmings and so on, I believe is about 10,000 British pounds, which could be like 12,000 euro, around in that range, yeah, more or less. But I can also tell you that the suppliers in different countries, they sell it at different rates. So if you buy it from low-income country, it will be cheaper. If you buy it from a rich country like the Trump one, it will be more expensive.
Okay, thank you.
My question is for Dr. Ragab. Can Cosmos UK distinguish, it's a very basic question actually, but can Cosmos UK distinguish between soil moisture changes driven by external inputs, let's say for example rainfall, versus internal changes like let's say for example compaction of structural alteration, etc. of soil. So can Cosmos UK make that changes?
Yes. So what actually you mean here? Would the Cosmos reflect the changes?
Exactly. Can it make the changes between?
Yeah. Well, I think most of people actually not interested in the absolute value of Cosmos. I can tell you this. They are more interested in the changes over time due to management. And there was one trial from my group to look at one interesting thing. In the UK now, there is a trend they call it organic farming and zero tillage. And there was a research by a small group. They were looking at the soil moisture under zero tillage, organic farming, compared to the traditional. And because you don't really plow the soil and organic farming and zero tillage, and you add organic matter, so the water retention is more. So you can feel that. And I think that something you could actually try here, also in Laayoune. Yeah.
Thank you.
Any other questions?
Thank you for your presentation. My question is for Abdel Rasool. I'm wondering about what's the most critical biochar properties to improve crop yield under salinity and drought stress.
Thank you for your questions. If you mean that, I think I put some of the characteristics, but try to avoid any salinity of, and try to produce the biochar with low temperature, so even the pH will be lower, so you don't add a high pH to your calcareous soils. And also the salinity depend on the materials that you used or the residue of the crops that you use. And these are two factors that are very important in the biochar, at least to the best of my knowledge as a water conservator.
Thank you.
Thank you. Last question, please, and quick, please.
السلام عليكم ورحمة الله. سؤالي موجه للخبير.
دكتور رجب.
أتذكر اسمه، آسفة جدا.
دكتور رجب.
أولا أعتذر لكم، قدراتي لا تسعفني في التحدث باللغة الإنجليزية بعد، لكن الحمد لله على الترجمة التي تجعلنا نتفاهم فيما بيننا. سؤالي للخبير: ما هي أفضل الطرق والأدوات المستخدمة لقياس الرطوبة في التربة بدقة، وكيف يمكن استخدام هذه القياسات لتحسين إدارة المياه في الزراعة وزيادة كفاءة استخدام الموارد المائية؟ وشكرا.
Dr. You can respond in Arabic.
Okay, I'll try my best in Arabic. I hope there is a translation to the others. Okay, thank you. بالنسبة لاستخدام أجهزة القياس الأرضية بتعتمد على نوع التطبيق. هل الاهتمام أساساً بقياسات على مسافات بسيطة، ولا أساساً أنت محتاجة أنك تقيسي الاحتياجات المائية لغيط كبير؟ فلو أنت أساساً بتحاولي أن تشوفي الاحتياجات المائية باستخدام الرطوبة، في الحالة دي الرطوبة في حد ذاتها بنحاول ان احنا نحسب منها النقص الرطوبي، مش الرطوبة في حد ذاتها كقيمة فعلية. احنا بنحاول نشوف الفيلد كاباسيتي او السعة الحقلية، وبناخد المحتوى الرطوبي اللي هو بنقيسه، وبناخد الفرق ما بين السعة الحقلية والمحتوى الرطوبي، وده اللي احنا بنسميه السول مويستشر ديفيسيت او النقص الرطوبي. لو انت بتدوري على حاجه ان هي تساعدك في حساب الاحتياجات بتاعت الري لفدان او هكتار، في الحاله دي القياسات اللي هي في نقط معينه دي ممكن تديكي فكره، بس لازم بتاخدي قياسات عديده جدا عشان المتوسط يبقى ممثل، لانه في اختلافات كتيرة جدا في الرطوبة ما بين متر ومتر آخر. فبالنسبة لنا احنا أساسا معظم شغلنا في مجال الهيدرولوجي، بنستخدمه طبعا مساحات كبيرة، فالقياسات بتاعت نقط معينة ما تنفعناش في حسابات جريان السطحي. لكن بالنسبة للري برضه هذه القياسات بتاعت الكوزموس ممكن تنفع. ERT لو في إمكانية تستخدم ERT اللي هي Electro Resistivity Tomography، احنا استخدمناه ده في الري، فده برضو بيديكي مش فكرة فقط عن المحتوى الرطوبي والسو موشن ديفيسيت، لكن أيضا بيديكي نوع من التوزيع وكفاءة توزيع مياه الري على يعني على مسافات من 32 متر 64 متر ل 96 متر والى اخره. فدي برضه ده لو في الامكانيات الماديه تسمح بالكوزموس يبقى كويس، ما كانش الاي ار تي، ما كانش يبقى البروفايل بروب، بس طبعا لازم تاخدي عده نقاط قياسيه. هي بتعتمد على الامكانيات الحقيقه. ده اذا كنت بتدوري على حساب الاحتياجات المائيه عن طريق الرطوبه. لو عايزه تحسب احتياجات المائيه عن طريق البخر، يبقى الطرق الحديثه اللي انا كنت بتكلم عليها اللي السنتونومتري والايديكو فاريانس واللايسومتر. في بعض الناس بتقول ما عنديش الامكانيات الماديه. مش بالضروره يكون عندك سنتيلومتر او ايديكو فاريانس. ممكن الواحد يعمل تجربه بسيطه جدا كلايسومتر على سبيل المثال، ويقارن عن طريق الوزن، الفرق في الوزن، البخر ناتح من هذا اللايسومتر او الصيص بما يحسب من المعادله اللي انت بتستخدمها في المنطقه، ويحاول يحاول ان هو يشوف الكوركشن فاكتور ايه، ويستخدم الكوركشن ده فاكتور ده لحسابات الاحتياجات المائيه عن طريق البخر. مش بالضروره كل واحد يكون عنده سنتيلومتر او ايديكو فاريانس. انت عليك انك تعملي تجربه صغيره لايسومتر والصيص من الفخار حتى، بس تحسبي الفرق في الوزن نتيجه للبخر، وبعدين تشوفي قرني ده بقى المعادله اللي انت بتحسبي بيها الاحتياجات بتاعت المائيه، وتشوفي الكوركشن فاكتور او معامل التصحيح، وفي الحاله دي ممكن انت تحسبي الاحتياجات المائيه برضو بطريقه ارخص.
شكرا جزيلا. Thank you, Dr. Ragab. Thank for our speakers. With this, I think we finished this session because I think we're late. So I would like to thank all the speakers, and we'll go for the next session. Applaud to our friends. Thank you. Sorry, Dennis. We started late too.
Ladies and gentlemen, good afternoon once again. Thank you, Professor Chukur Allah, for that good leadership. So we'll move on to the next session. We have quite limited time. My name is Professor Dennis Sashlenje. So I'll lead the session number 6.4 about agricultural use of soil amendment, fertilization, and microbes association. We have 4 presentations scheduled to finish our day today. So without wasting time, I'll welcome the first presenter, Khalid Azim. I don't know if he's around, to speak about organic amendments to restore sandy soils to produce organic vegetables under arid conditions. 10 minutes, please.
Thank you very much. Well, I'm happy to be here and for the second time, and I'm happy also to share some results, insights about scientific findings about using organic amendments in soil to fertilize or to amend the soils grown where we grow organic vegetables crops. So maybe you know, first of all, I have to present myself. I'm Khalid Azim, researcher in organic agriculture and also researcher in organic waste management through composting, vermicomposting, and so on. Based in Souss-Massa region, maybe you know it. Souss-Massa region is the most productive region of tomato to export, local consumption, also for export to EU. And we have intensive way of cropping this crop, tomato, and we have too much waste generated with this crop. It's kind of intensive way that we have lost our quality of our soils, sandy soils. We have soil depletion. We have arrived to raise, or maybe to have 5 0.5% of organic matter is quite mineral soil. And also we have some soil-borne pests and diseases like nematodes, and many fungi and bacteria that are affecting our cropping system. High fertilization rate and low efficiency, I don't know, hopefully or unfortunately we have this rise of price of fertilizers, so maybe farmers started to decrease the rates of fertilization rate. And also we have this groundwater contamination with fertilizers and also with pesticides. Water overexploitation is common. We have used more than 10,000 tons per hectare per year for growing tomato crops, but now we arrive to use only 6,000 tons per hectare. And also we have used more, and not in a manner, but not in a logical manner. We have used organic matter, organic amendments with high rates, using 30 to 80 tons per hectare per year, which was very excessive. And I think also with the rise of the price of compost, now it's quite 900 dirhams per ton. It's 1,000 tons. 1,000 dirhams per ton is 100 euros per ton. Compared to 20 euros in Europe, it's very expensive for a farmer, for a regular farmer to use compost in his organic production or conventional production. So the question was actually what to amend, what kind of compost we could amend to the sandy soils, and also how much rates. 80, 30, 10 tons per hectare, or maybe nothing. And the third question, for how long are we going to amend once a year, once in 2 years? What is the frequency of organic amendments in organic vegetable production? So all those questions were raised, and we tried to answer those kind of questions. And also, that was one of my questions raised during all the 6 years of cropping organic vegetables. So our objective is to evaluate the midterm impact of compost on soil fertility, also monitoring the mineralization kinetics of all the nutrients in soil, and compare the 4 rates of compost amendments. We have used, I don't know how, yes. The first year was squash, the second year of confirmation of the results, green bean as a legumes, melon and pepper for the 5th year. And the 6th year was quite a fallow year. So the 4 rates were used, 10, 15, 20, and 25 ton per hectare per year, in addition to the control no-compost amendments, to compare what are the benefit that we're getting with organic amendments in soil. 4 depths were assessed, 0, 30, 30, 50, 50, 70, and 70, 90. The question actually was that after every amendment, we had 0.5% of organic amendments, and after the first amendment, we got our organic amendment rate content of 3.7%. At the end of the cropping season, we are left with 1.5 or 1.7%. Where is the deficit is coming? Where the deficit is going, actually. And so we were questioned, maybe those organic materials, mineralized or maybe decomposed, were, how to say, disloced, or I don't know how to call it. Maybe we have to look on them, to look for them in the depth. So we use an auger to measure, to take soil samples. We have analyzed the pH, EC, soil organic methods, all the major nutrients analysis in the soil. So now we got to the results. As you can see that organic amendments after 5 years of cropping system could make a good result, on decreasing the pH of the soil. And this is a quite stable level of pH soil compared to the control. And we can see also that the pH of soil has been decreased compared to the control in all the layers, in all the depths in the soil. Concerning the soil EC, the conductivity level, also we have observed that there is an increase compared to the control in all the layers. The cation exchange capacity also has been affected by organic amendments in different rates. we could see that it has been observed an increase in CSA in the topsoil and also in the other layer. That means that we're going to see it in the organic matter also, that we have accumulation of organic matter in the other layers. That means that organic matter, mineralized or not, are dislocated to the other layers in the depth. Available phosphorus also has been affected by organic amendment, but not in the topsoil. In the second layer, we could see that there is an increase with increasing compost amendments in the fields, but not here, because we have exportation, plant uptake from the soil. Concerning the human content, we could see that there is an increase also with increasing rate of compost compared to the control, and also in other layers. The exchangeable potassium also the same, but we could see that here there is an accumulation of potassium in this layer, which could be how to say a good indicator that we have to look for crops that have soil roots deeper than 30 centimeters. Fulvic acids also, we have accumulation of fulvic acid in the top layers, but not here. But here with the 25 ton per hectare of compost per hectare, we have accumulation of fulvic acid in the 90 centimeters depth. Humic acid also have been increased in the topsoil, but not in the second, in the third and the fourth, but also here in the second one. So we can see that after those results, I didn't present all the results. We have also some results related to minor elements and minor elements and oligoelements that hasn't been presented here. We could conclude that compost amendments has improved the soil fertility at the top layer of 0 turns, especially with the rate of 20 and 25 ton per hectare, and also the improvement in cation exchange capacity compared to the control, with better availability of microelements to crops. Leaching of organic acids could be a relevant indicator that we have to look for crops that have deep root zone to explore what we couldn't do with the top layer. The microelements, copper, iron, and magnesium, manganese has been leached and should be taken into consideration in the cropping system. We recommend that to use only 50 ton per hectare of compost in organic vegetables, and the tonal crop, it's better than using 25 because we reduce the price and we have the same results. For maximum nutrient efficiency and recovery, fruit trees should be explored in this cropping system. And also, I'm coming to the comment to the conclusion of my colleague from Saudi Arabia that say that we have to add clay, and that was one of my recommendations, to have an annual low clay amendments in addition to the compost, to make this complex of clay humic complex increase in the soil and have more how to say nutrients balance in the soil rather than having them leaching. Thank you very much.
Very good. Thank you, Mr. Zim. So you'll keep your questions until the end of the session. So we'll move on to the next presenter, Barak El Gouria, PhD student, and he's going to talk about combined effect of irrigation regimes and NPK fertilizer rates on soil health, maize performance in marginal lands. Welcome.
Thank you, Professor Denise. Thank you so much for your presentation and presenting me to the colleagues who are attending my presentation. Hopefully that you are having a good evening with us, and you are very welcome to the conference and to the middle of the deserts. You are welcome to Asari. So I will start my presentation as it looks from the first slide. I've changed the background, so I'm working on another crop, not blue panicum, so I changed the background. It's maize, it's corn, silage corn. So my presentation is entitled on combined effect of deficit irrigation, but after the presentation that Professor Ragab has given, it will not be like deficit irrigation because we worked using the Piemonte-Montiet equation. So as he talked that this equation overestimates the water requirements. So and then we will see the combined effect of this deficit irrigation that we used and the different NPK rates on soil nutrient status and soil properties, salinity, and also on silage corn properties and performance and productivity under saline conditions. So to move into the second slide, it will be on, especially on a key context that I would like to highlight based on the statistical calculations that have been done by the FAO. So as you all know that the marginal lands, it's almost 38% of 21% of the total global lands that we do have, which is almost 2.74 billion hectares, and 1.56 billion hectares are under agricultural use. This is globally. And also, among this 156% of the soil, between 200 to 300 million hectares are agriculturally marginal. So we are using the marginal lands to grow crops due to the insufficient food supply that we are facing. This leads us that almost 38% of the population is living in these regions, which means that 1.6 billion of them are under developing in developing countries. Also, the second pillar is that the poverty and food security that we are facing. So mainly 734 million of the people are living with less than $2 per day, which means that 820 million of them are undernourished. This leads that the marginal lands users are highly vulnerable due to the environmental changes and climate change. The third point is that the recent pandemic that we had leads to different and several impact on the society, mainly the worsened the food supply to the different populations in different regions. Also, it delayed the progress toward no hunger or ending hunger worldwide. The third key point is that it pushed 83 to 132 million people to food insecurity. This leads us to the goals that we set for sustainable developments at risk, mainly ending zero hunger and no poverty, which needs an immediate action mainly related to sustaining productivity and using generative agriculture to sustain and enhance food supply to different communities wherever they are. And also the second point is that we should boost the agricultural productivity, especially using the marginal lands that we do have, as the agricultural lands are starting to be depleted. By this, I move to, we defined two main objectives with our one aim, which is to study and investigate the combined effect of the irrigation regimes that we used and the amendments that we applied, mainly the NPK rates, on soil health indicators and also on maize growth and performance in the result-affected areas. The main objectives were evaluating the effect of this deficit irrigation on soil properties and also investigating and evaluating the effect of combined deficit irrigation and NPK fertilizer rates on our crop that we are growing. So the methodology that I have used is that, by the way, the experiment was established last year in Phosboucraa region near Laayoune city, so in the place called Jaradi, with the farmers. So we are working with farmers in their farms. So they give us a piece of land of their farms, and we are working together with them in order to find better solutions for their farming activities. So it was set as a randomized complete block design with a split plot arrangement. The main plot were the irrigation regimes. We used 100% and 50% of crop evapotranspiration, and also the amendments were from 0 to 240 kgs per hectare. This was replicated 4 times. I would like to mention here that the land that we have used, it wasn't used for too many years. So we try to apply a standard dose of compost, which was 4 tons per hectare, before starting our experiments. So by this, it's like our control was not a zero. Our control was the 4 tons per hectare of the compost. Moving now to the result that we got. So for soil properties, especially for the potassium levels and for sodium levels, as are the main nutrients related to the salinity. So we have found some significant variation in terms of potassium levels under the control and also the treatment that we have applied. However, our control, which was the 4 ton per hectare of compost, shows higher levels of potassium in the soil compared to other treatments. With 120 kgs of NPK shows higher compared to the different other NPK doses. However, for the sodium, it didn't show any statistical significant variation. However, the sodium concentrations was similarly for all the treatments. So now moving to the physiological parameters of our crop that we have studied, starting with the proline concentrations at 2 growth stages, were mainly V2, V1, and then moving to the reproductive stages, which is the first one, early reproductive stages. As it mentioned here by this slice, the proline concentrations at the V1, the V12 stage shows higher proline concentrations under well-watered conditions, which is decreased under deficit irrigation by almost 26%. But at the R1 growth stage, it's like the difference. It starts to be it decreased under well-watered conditions, and it's higher in deficit irrigation regime, which means that our crop is starting to produce proline in order to tolerate, in order to adopt its mechanisms to the salinity levels that we are applying. Also, for the relative water content, it shows the relative water content showed different. Okay, nice. 3 minutes. Okay, it did show us some significant variation, which was minimal. It wasn't a high difference between the well-watered conditions and the deficit irrigation. However, the second figure here, it shows that the relative water content starts to increase with go into the late growth stages. For the electrolyte leakage, it didn't show any significant variations, but at the R1 stage showed higher levels of electrolyte leakage, which means that at this early reproductive stages, crop starts to use water more than in the other stages, which increased the salts in the leaf, the crop tissues. Now moving to the agronomic parameters, unfortunately we didn't get any significant differentiations for the measured parameters. However, for the canopy height, our crop reaches 1.6 meter. However, for the leaf area, it was around 400. Now for the yield, also it didn't give any significant variation, but for the green yield, it was almost between 4 to 5 tons per hectare, and for the dry yield we reached 17, which is a little bit less than the one obtained in normal conditions. So by this, my conclusions are enhancing the irrigation management is crucial to increase irrigation water use efficiency and also subsequently fostering agricultural sustainability in marginal lands. Soil potassium levels were significantly varied among the treatments, with slight decrease under NPK rates with the control, which was 4 tons per hectare of compost. Also, the leaf proline content showed a decrease between V12 to R1 growing stages under well-watered conditions, and increased under the deficit irrigation. The fourth point is that the maize dry biomass yield obtained was almost 17 tons per hectare under these salt-affected conditions. And the last point is that we still have to do some further research in both in agricultural activities and mostly sustaining crop production in salt-affected areas here in our southern part of Morocco. So by this, I would like to thank you so much for your attention, and that's it. Thank you.
Thank you, Marek. Very concise. So we'll move on to the third presenter, Khaled Al-Taibi. Hope I got your name right. Impact of integrated use of mineral fertilizer, composition and irrigation with treated water of fish.
Thank you. Yeah. Assalamu alaikum. First, I would like to introduce myself. My name is Khaled Al-Taibi. I'm Associate Professor of Soil Fertility and Plant Nutrition at Soil Science Department, King Saud University. It's my pleasure to be here and to share with you the findings of our research studies titled The Impact of Integrated Use of Mineral Fertilizer, Compost and Treated Wastewater and Crop Yield and Nutrition in Arid Soil of Saudi Arabia. I'll just go through a brief introduction background about the issue of the water. The water scarcity is a big issue in arid region and semi-arid regions, and this is because of the hard environmental conditions, low rainfall and high evapotranspiration and hot temperature, and this threats the crop production sustainability. Saudi Arabia is located within arid regions, and it's characterized, the weather is characterized by high temperature, low rainfall, and also this results in limited water resources. And this water shortage is also worsened by some activity, agricultural and industrial activities. So during the last few decades, there was an expansion in agricultural production in the countries, and this results in depletion of the groundwater, which is the main source of irrigation water. Also the industrial development and also rapid population growth, and there was a lack of a water conservation culture in previous years. These factors results in some measures taken by the government, and this includes suspending the cultivation of large-scale green forage crops, and also there was some alternative approaches have been taken during the last few years. This includes introducing low water consuming crops and also implementing new agricultural methods that can enhance irrigation efficiency. This also includes reusing treated wastewater in agriculture. This statistical information is showed that water consumption or use in 2016 in Saudi Arabia. So it was about 25 billion cubic meters, and it distributed between these sectors. Mainly, you can see here 20 or 82 of this consumption is by agricultural use, or it's through agricultural activity. And 90% of water sources are coming from groundwater, which is not renewable. And it's expected or estimated that the agricultural water use will reach about 25 billion cubic meter by 2030. However, this has been accompanied by promoting the use of treated wastewater and for agricultural irrigation. So at a global level, there is, in the same in arid and semi-arid regions, there is increasing use of treated wastewater and agricultural irrigations. And, but national in Saudi Arabia, or at the national level, the amount of treated wastewater used in 2015 was about 0.61 million cubic meters per day. That's by all sectors. And majority of this use is by agricultural sector, which is about 0.40 million cubic meter per day. And the national water strategies includes promoting and expanding the reuse of treated wastewater in agricultural sector. And it's expected that by 2030, the treated wastewater use will reach about 16%. So instead of using as an irrigation source, treated wastewaters also contain considerable amount of nutrients, plant nutrients. And if it's used for irrigation, it also can provide nutrients which can partially replace the mineral fertilizer. And it can reduce reliance on chemical fertilizer. And there are many studies documented globally, but at the local level, there is limited research studies examining the impact of integrating treated wastewater used with mineral fertilizer or compost under the arid soil conditions of Saudi Arabia. So there are some, we have created greenhouse studies and also field studies. And the main object of the field study is to look at the impact of integrating mineral fertilizer and treated wastewater and compost on sorghum and barley yield and nutrient content. And also under greenhouse conditions, we look at the interactive effect of started bee fertilizer rate and treated wastewater and early growth of corn and uptake of phosphorus. This is the experimental design of the greenhouse. There is several treatments, 4 rates of phosphorus and 2 types of phosphorus fertilizer, DAP and SSP, diammonium phosphate and single superphosphate. And we use treated wastewater and tap water. And we grow maize for only 6 weeks. For the field studies, we conducted a field study, and this is the location. You can see it in the map. This is the agricultural experiments and research station, belongs to King Saud University, just south of Riyadh. And you can see the red square. This is exactly the experimental site where we carried out our experiment. And we used treated wastewater and fresh water, and we used 5 rates of mineral fertilizer. And also we used compost at 3 rates. And it was for 2 seasons. 1 season is for barley and the other season is for sorghum. This shows the irrigation design. As you can see, 1 tank is for treated wastewater. The other one is for fresh water. And this is the plot layout and the irrigation design. And it shows the lines of water. And the experimental plots is 2 by 2 meter. This is the basic characteristics of the study from the experimental site. As you can see here, the soil is poor in organic matter, it's alkaline, and also it's poor in nutrients, and it's dominated by a sandy loam texture. And this is the treated wastewater basic properties. As you can see here, it's contain considerable amounts of nutrients. This is the results from the greenhouse. I hope you can see the x-axis, the labels. And this is the plant height, the root length, and the dry matter yield. And overall, the treated wastewater promoted the root length and dry matter yield, and so plant height comparing to the tap water. Also, the B content increased under the use of treated wastewater, and also the nitrogen content, phosphorus content, and also potassium content, comparing to the irrigation with tap water. For the field studies, this shows the impact on barley yield, either the total biomass or the grain yield. Generally, treated wastewater promoted crop yield and increased the yield comparing to the irrigation with the fresh water. And there was inconsistency between the rates of fertilizers, especially the compost. And this is average. Okay, this is average across the fertilizer treatments or the other treatments. So you can see here the treated wastewater and fresh water. And the treated wastewater increased the yield by about 25% for both for biomass and grain yield. And this is the mineral fertilizer. So the yield increased with increasing the fertilizer rates. And this is the nutrient content in barley. It's followed almost similar pattern as in the yield. And that was for the nitrogen. This is for phosphorus. It shows similar pattern of the impact. And this is the effect on sorghum yield. And sorghum yield, we did not include the compost because it's not showed a clear impact, and also on some plots it's inhibited crop growth. So we just focused the mineral fertilizer interaction with the treated wastewater. So it's clear here the treated wastewater increased the yield comparing to the fresh water in the right for total biomass or for the green yield. And also for the plant nitrogen content and for plant phosphorus contents, the nutrient content increased significantly with the use of treated wastewater. And this is also for the potassium. Also, it increased the content of potassium in treated wastewater compared to the fresh water. So the conclusion is that, as you can see, the treated wastewater promoted crop growth and yield, and the started bee fertilizer efficiency was enhanced with the use of treated wastewater, and the crop yield under the field conditions was also increased. And one reason it can be treated wastewater improved the nutrient efficiency under when the treated wastewater was used, as in this soil conditions, the nutrient use efficiency is very low, especially for phosphorus and nitrogen. So the treated wastewater has a potential to partially provide plant requirements for nutrients and reduce dependency on chemical fertilizer use. And one possible reason is that the treated wastewater contains soluble nutrients and organic matter, and this can promote soil microbial activity and organic matter turnover, and this will enhance crop growth and development and nutrient utilization. However, from this short field study, we cannot reach a final conclusion because using treated wastewater needs a long-term experiment to make sure that there was no negative impacts on soil or environment. So this requires long-term continuous application of treated wastewater to look at its effect in soil fertility and accumulation of nutrients in soil, and also microbial activity. And thank you very much. Thank you.
Thank you. Thank you, Dr. Khalid. So next, last presenter, Fadwa, please come. She will talk about optimizing tomato resilience to soil stress using compost, Phosboucraa mycorrhiza fungi, and their synergistic effects. Please be very fast, 10 minutes only. Welcome.
So, hello everyone. I'm Fadwa Mkawi, PhD student, and I'm so delighted to be here today to present my results about my research topic entitled Optimizing Tomato Resilience to Cell Stress Using Compost, Phosboucraa mycorrhiza fungi and their synergistic effects. So, as you may know, climate change is indeed a major problem that has consequences on the environment and human societies. It can be of natural or anthropogenic origin, and it can manifest itself in the form of dermal stress, water stress, metal stress, and saline stress, which is the focus of this study. So, the saline stress has a several negative effects on plants and the soil. It can cause a decrease in fertility and soil degradation, significant reduction in yield and slowing down on growth, and it also reduces chlorophyll content, which affects photosynthesis and sugar production. It disrupts the plant's ability to absorb water and nutrients necessary for its growth and decreases leaf retention and plant's height. So with an ever-growing world population, the use of saline water and soil will become inevitable. Therefore, it is crucial to develop new sustainable and environmentally friendly management for the use of these valuable resources. So the objectives or the aims of our studies is to evaluate the effects of compost and Phosboucraa mycorrhiza fungi applied individually or in combination on tomato tolerance to salt stress, to analyze the morphological, physiological, and biochemical responses of tomato plants subjected to different treatments, and to identify an effective biostimulation strategy to improve growth, photosynthesis, and the resilience of tomato under saline conditions. So the plant materials used in this study comprise tomato plants of Campbell 33 variety. Various treatments were applied, including control, compost treatments, Phosboucraa mycorrhiza fungi treatments, and their combination. And saline stress was induced at two levels, 0 millimolarity and 115 millimolarity. The compost was applied at a rate of 5%, and the mycorrhiza was obtained by multiplying them in maize plant that promotes proliferation, and then were applied at the root at a rate of 20 grams per plant. And subsequently, a series of analyses were conducted to assess the effects of treatments and salt stress. So now we move to results, and there is some numbers. So salt stress cause or reduce a tomato grow by 17% in plants high, 13% in number of leaves, 58% in number of fruits, 32% in shoot fresh whites, 65% in root dry whites, and 10% in photosynthetic efficiency. While in contrast, biostimulants improve these traits. Phosboucraa mycorrhiza fungi improve number of leaves by 15%, and number of flowers by 429%. And the combination between Phosboucraa mycorrhiza fungi and compost improved number of fruits by 125%, and all biostimulants improved the photosynthetic efficiency by 7 to 12%. So the results showed that no colonization in control plants was observed, and compost reduced mycorrhiza parameter under salt stress, while salt stress increased colonization intensity in Phosboucraa mycorrhiza fungi treated plants by 38%. So salt stress reduced pigment content by 36% in chlorophyll A, 6% in chlorophyll B, and 93% in carotenoid. And biostimulants mitigate the negative impact of salinity and increase total chlorophyll A and B in concentration. And the best results was with M treatment or mycorrhiza treatment. So salt stress reduced also nitrogen content by 38% in leaf nitrogen and 8% in root nitrogen content. And biostimulants increase nitrogen levels in both leaves and roots compared to the stressed control. And the highest treatment, or the highest value was recorded with the mycorrhiza treatment and the combination treatment. So saline stress triggered the oxidative response by increasing hydrogen peroxide levels by 76% in leaves and 108% in roots, and MDA by 228% in leaves and 117% in roots, while the biostimulants reduce reduces these stress markers. In leaves, hydrogen peroxide decreased by 16% in mycorrhiza treatments and MGA by 56% in mycorrhiza. And in roots, hydrogen peroxide levels decreased by 79% and MGA by 18% in mycorrhiza treatments. So under salt stress, soluble sugar increased by 278% in roots, and no significant change were observed in leaves. And biostimulants increased these levels. In leaves, the compost increased by 217% and mycorrhiza by 146%, and the combination by 214%, while in roots, compost improved by 29% and the combination by 168%. And also salt stress decreases protein content, while the biostimulants increase this content. So for the catalase activity in shoots, salt stress increased catalase activity by 143, while biostimulants just with 53% compared to stress control. And no significant effects of stress or biostimulants was observed with the ascorbate peroxidase activity. And in roots, okay, it's the last. And in roots, only C treatment improved the ascorbate peroxidase level under salt stress with 132%. So I will just conclude with the last PCA results. Under non-saline conditions, compost and or Phosboucraa mycorrhiza fungi treatment showed strong positive correlation with growth parameter, number of leaves, and others, and nutritional and pigment traits. And these parameters were negatively associated with salt stress, while salt stress was positively correlated with oxidative stress markers. And under saline conditions, Phosboucraa mycorrhiza fungi alone or with the combination with the compost showed strong correlation with the number of leaves, number of flowers, root elongation. And compost under salt stress was positively associated with root sugar content, antioxidant enzyme, fruit, and photosynthetic efficiency. And I think I conclude with the others. So thank you for your attention.
Thank you, Fadwa. So time is really gone. I guess it's time for tea. But I'll just allow one question only. Just one. Just one. Any question? A quick one? Yes, Professor Testa.
I could ask a quick one. I enjoyed all of these talks, actually. So thank you to all of the speakers. And I have a question for all of the speakers, which is whether, or perhaps it's more encouragement than a question, but it would be great to see if you are able to, not just in inverted commas, present the results of increased plant growth, increased yield and so on, but also the cost-benefit analysis for the farmers. So these treatments cost money. If you're able to provide estimates of that, and if you're able to provide estimates of the benefits to the farmers in terms of increased yield and thus increased revenue, because we really have to be starting to work towards more than just in inverted commas a description of these types of responses, but also how they can really be turned into a business case for the farmer.
Thank you.
Maybe you've all done that already, but
Probably is in pipeline. Thank you so much. Thank you, Professor Tester. So, just because of time, we have exhausted our time. What I would recommend, to be fair, is that we have the time for tea. We can discuss, but I want us to give an applause to our presenters for the very good presentations. That was really interesting talk. You are welcome. Thank you.